Good morning to the day 2 of the OCB workshop. And before I start, how many of you are sitting in your old seat from yesterday? All right. Let me introduce the team, dream team. My name is Xinping Hu, and my co-conspirator, my co-chairs, Emily Osborne and Matheus Fagundes. Matheus is a PhD student from UGA, so I did my postdoc at UGA, but we're not related. Okay. All right. So this morning's session is Marginal C biogeochemical cycle in the Anthropocene. And first of all, what is Marginal C? This is definition, I came up operating, it's not invented. But yesterday there was discussions on the AI. All right, so a few weeks ago, I just thought, what about ask AI to make a cartoon of the marginal Cs? And I tried a few different platforms. So this is a prompt. Okay, I define what marginal C is. If you have seen the famous painting of the melted clock, okay, Stable diffusion, that's like my daughters when they were like 7-year-old painting. And the best one is Mid-Journey. Actually, I paid the subscription and, got a few down. All right. So I don't know, if you guys want to, I think they will improve in the future. Well, one thing I forgot, I specified no animals. You can see, you can find animals, in all images. All right, so the characteristics of marginal Cs. In general, they have large shelf to basin ratios, a lot of shelves, smaller basin, shallower depth than the open ocean, and a lot of terrestrial influence, for example, Gulf of Mexico, East China Sea, big rivers, and also Arctic rivers. That's the most changes are happening there. And there are water mass implications. So marginal Cs are involved in, either producing water masses or water masses getting into the marginal Cs. And boundary currents, they are affecting marginal Cs, some of them. And of course, very large changes because of climate change and anthropogenic activities, they are altering marginal Cs as well. But one question I think we can think of is whether we could use marginal Cs as analog to study the open ocean.
So our objectives of this session this morning are really to bring an interdisciplinary group of speakers together talking about a range of marginal C settings. And we've asked our speakers to synthesize the state of the knowledge with respect to biogeochemical cycling across these different systems that have many different influences and dynamics, and describe how these cycling dynamics may be varying as a result of anthropogenic change and inputs. And lastly, we've asked our speakers to draw connections between biogeochemistry and marine ecosystems, coastal to open ocean interactions, as well as we have a little bit of a flavor of marine carbon dioxide removal research this afternoon. Will be a continuation of that discussion. So to set the scene a little bit for you and what to expect this morning, so we're going to kick it off with some more targeted topical science talks. These are going to be 15-minute talks. So Nina is going to start us off with a paleo perspective, go geology. And then we're going to have Hongjie coming in and talking with us about a management case study focused on nutrient management. We'll have Mona coming in and talking with us about the benthic part of the system with a focus on alkalinity. We'll have Brenda Carter talking with us about anthropogenic impacts in marginal C settings, and we'll wrap it up with our first kind of part of the session talking about considering applications of MCDR and marginal Cs with a specific case study that was applied in the Northern Sea with Sophie. We're going to have all of our speakers come up and from this morning part of the session, and we'll have kind of a group Q&A session as we're transitioning between talks. If there's a question, we can certainly take that, but we're going to try to hold questions to the end. So hold on to those questions that you have as you're listening to these talks. think we'll take a quick break there, and then we'll come back and we'll have slightly longer. So we'll have 20-minute talks, which are focused regionally, and these are going to be more kind of big picture synthesis talks. We'll have Marta talking with us about the Mediterranean. Hongjie is going to talk about the Arctic. We'll have Katak Lee talking about the Western Pacific marginal seas. And rounding out the session, we'll have Andrea online talking with us about the Gulf of Mexico. Again, we'll have another Q&A panel with all these speakers, and then we'll bring everyone back up at the very end, and we'll have a panel discussion to round us out before lunch. And so with that, you've probably seen this picture a lot, and we're hoping that you found your home in your marginal sea. You're going to be at the cheering section for our speakers. So these are the marginal seas that are featured within our session today. And without further ado, I will hand it over to Matt, who's going to be introducing our speakers this morning.
Hello, everyone. I was just standing there and I was like, oh my God, it looks like I'm in high school standing with everyone, ready to present. Okay. So our first speaker it's Nina Papadomanolaki.
First of all, thanks very much for having me. It's my first invited talk, so I hope it goes well. I just want to apologize if during my talk you hear any thunder sounds. The weather is a bit bad, but I think it's not going to be too loud. So as was already mentioned, before we dive into the marginal sea, biogeochemistry and dynamics in the Anthropocene, I first want to give a bit of a perspective for the role they may have played in the geological past, and specifically during the Paleocene-Eocene Thermal maximum, which was a climate change event about 56 million years ago. I'll start with giving a bit of an introduction to organic carbon burial, which is the process that I'll be focusing on. So briefly, what the role of marginal seas is, what kind of processes control organic carbon burial, and what the future may hold, what role it may play, before moving on to the Paleocene world, where I'll give a short introduction to the PTM itself, which is the event I'll be talking about, and then a couple of case studies for two specific marginal seas, and then a more global overview for what we know about organic carbon burial during the PTM, both from data and from some modeling work. So it was very nice to see what the definition that we're going to be using for marginal seas is. However, when going through papers, I've got the feeling that some terms are used interchangeably, especially in the paleo community. So I think I'll be covering all of these. In some cases, the terms may be used for the same thing. So coastal ocean may be used for a shelf sea or a marginal sea. In other cases, they may be a bit separate, but I'll try to stay on topic. So organic carbon is, and its burial in particular, is pretty important in the global carbon cycle because it's one of the long-term sinks. So after exchange between the ocean and the atmosphere system, organic carbon burial and weathering of terrestrial rocks are the main mechanisms that remove atmospheric CO2 from the system. So CO2 dissolves into the surface ocean, where it can be used up for the production of organic matter. A small fraction will escape degradation in the surface ocean and be exported into the ocean interior, but an even smaller percentage of that will eventually be deposited on the seafloor. And then over time, after some diagenetic processes and other benthic processes have taken place, a very small fraction is buried for the really long term. However, it's that small fraction that actually can play a quite big role, because the CO2 that is captured in the organic matter that's been buried is going to be removed from the system for millions of years. Now, most of the burial happens on shelf and in marginal seas. So most of it, or a bigger fraction of it, is in deltaic regions. But outside of that, it's still a very large amount of carbon, especially if you think about what small fraction it is of the original production. And especially when you compare it to the open ocean, which is just a tiny, tiny amount, relatively speaking. Of course, marginal seas and other such coastal areas are quite dynamic environments. So even though as a whole they play a large role in organic carbon burial, it can be quite localized. So in the North Sea, for example, production takes place everywhere, but most of the burial only takes place in the Norwegian Channel and the Skagerrak near Denmark. In the Gulf of Mexico, some regions are more affected by terrestrial organic matter input, the ones that are generally closer to land, whereas the more open places are they bury more marine organic carbon. Now, there's a number of factors, of course, influencing organic carbon burial. I'm not going to go into all of them, but of course, the availability of nutrients in the surface ocean determines how much organic carbon can be produced. So more nutrients means more production, and if all else stays the same, then you would also get more burial. Exposure time to oxygen is important. The longer it's exposed, the more oxic degradation can take place. So at very long exposure times, you bury very little, whereas at short exposure times, you get much higher burial efficiency. And high sedimentation rates also favor organic carbon burial, as the deposition and also burial itself speeds up. So in the future, we know that some of these processes that control organic carbon burial are going to be changing. So oxygen is decreasing in the open ocean and also in the coastal oceans. In the coastal regions, it's due to nutrient loading a lot. In the open ocean, it's more due to warming. And we know from some modeling experiments that when you increase the nutrient input and you also simulate hypoxia at the same time, this can stimulate a lot of burial in marginal environments and potentially also in certain regions of the open ocean. And this organic carbon burial in some tens of thousands of years, so we are talking definitely long-term, will match the amount of CO2 that humans have emitted into the atmosphere. And at that point, they will start probably controlling the lifetime of our emissions in the atmosphere. So for the next, let's say, few centuries at least, we're not going to be looking at these processes much, but after that, organic carbon burial will be determining partly how much CO2 will stay in the atmosphere. So for the paleo event that I'll be talking about, we're going to move back quite far in the past. So this is the last 66 million years. At the bottom, we have the extinction of the dinosaurs. And the PTM is located quite soon after that, 56 million years ago, when the world was a lot warmer. So CO2 was a lot higher. We're talking about probably more than 1,000 ppm compared to today's amount. Temperatures were a lot higher. And sea levels were also a lot higher, resulting in this rather changed global outlook. So we've got very large areas of the continents that are basically drowned basins. So the shelf, and this includes marginal seas, was 25 to 50% larger than modern, which creates a lot of areas where this organic carbon burial could have taken place. Now, when we're talking about something that happened 56 million years ago, of course, the toolbox that we have is very different to the modern ocean. So we cannot measure processes directly. We have to measure what influence they had on different elements, on the deposition of specific materials like organic carbon, calcium carbonate, etc. And uncertainties are therefore quite large, as multiple processes can imprint on the same element or the same change in the sediments. And also we are very restricted by the deposition and preservation of sediments, as the coverage of suitable sites is quite limited. I mean, 56 million years is a lot of time for geology to do its thing and start removing a lot of these sediments from the record. So in some places, an event like the PTM, which lasted for 200,000 years, can be a couple of meters long. And in other places, we might also only have like 10 or 20 centimeters of it, which then limits very much in what kind of detail we can study the event. Before I'm going to talk about organic carbon burial on a global scale, I'll talk a bit about the North African Shelf Sea and the Paratethys, which was one of the largest epicontinental seaways at the time, which are two marginal basins that we have significant larger amount of data for than some of the other that you may see on this map. So starting with the North African Shelf, this panel was supposed to show later, but we have 9 sites in total. Most of them are in Egypt, so the spatial coverage isn't great. But all of these sites tell us that there's low oxygen during the PTM. So as CO2 emissions and methane emissions caused global warming and this very rapid climate change event, oxygen on the North African Shelf decreased. Most sites show either sustained high productivity or an increase during the PTM. But only 2 of those actually have high organic carbon content. And they're the ones that have the most reducing, so most low oxygen conditions of all of them. One is the one that you can see here. So the gray bars is the PTM event itself, which is marked by an excursion in carbon isotopes. This is one of the markers that we look for in the sediments. you can see that there's a very large increase from less than 1% sediment weight of organic carbon to almost 4.5%. And at the same time, all of these redox sensitive metals are increasing as well. So they're all showing that they were deposited under low oxygen conditions. And the time is more or less equivalent to when this organic carbon increases. And at the other side, we have also a record for nitrogen isotopes, which they can indicate a number of different things. But here it's been interpreted to show that there was a lot of productivity by cyanobacteria, which are a species that today blooms a lot in, for example, the Baltic Sea. And that there was also nitrogen fixation going on, partly because of the low oxygen conditions that were removing nitrogen through denitrification, and also because the nutrients were simply being used up very quickly due to organic matter production. So it seems that this high TOC is due to both the low oxygen and the high productivity. Then for the paratethys, we have a bit more coverage. So we are looking at 11 sites, which for an event from the geological history is quite a lot. And they're also spaced quite widely apart. So they cover different parts of the basin better than for the other basin that I showed. Again, most of them show us that there was either sustained low oxygen or a decrease. There's not a lot of data on productivity, but for all of the sites that do have some data, they all show an increase in productivity at the same time. And most of these sites do have a very high organic carbon content, which suggests that burial of organic carbon or the preservation once it was deposited on sediments was quite high. So for this one, for the southeastern part of the basin, people have developed a bit of a model of what it looked like. So this is before the PTM. We have 3 sites, 2 of which were located in a low oxygen zone, and the third one was still oxygenated. Then warming and sea level rise caused an expansion of this low oxygen zone, an influx of terrestrial material with lots of nutrients, and all of these sites start having higher organic carbon content. But then as sea level dropped and climate recovered, the low oxygen zone retracted as well. So 2 of the sites are now more or less outside of it, and only the third one remains inside. So just like modern marginal seas, it's a very dynamic environment even before you start adding rapid climate change to the equation. Now, we've done some compilation work on both the factors controlling organic carbon burial and organic carbon burial itself. Again, we are kind of restricted by the sort of data that we have. Ideally, we'd want to have information on rates of deposition of organic carbon, but those are not always easy to come by. This was a global compilation for high productivity and or deoxygenation. The circle here shows where most of the shallow marginal sea and or shelf sites are, and almost all of them show evidence for either increased productivity, so that's the redder colors, or a decrease in oxygen. And in many cases, you can see this dark red color, which means both of those processes were going on. We also find evidence for this in the open ocean. You cannot see that on this figure, but we have quite a few open ocean sites. But then when we look at organic carbon content in the sediments, almost all evidence for high values are in marginal seas and on the continental shelf, especially if we're looking at so-called black shales, which have a content of more than 2% weight, which is a lot. And for a lot of the open ocean sites, they're not even on the map because organic carbon content wasn't measured. This is not because people didn't have time or money to do it. It's just that you could see that it's going to be so low that it's not even worth measuring. So this really means that also in the ancient world, organic carbon burial was mostly a more shallow water process. And from that, we try to see a bit what its impact would have been on the global carbon cycle and on climate. So on the left, you see again that carbon isotope signature. What it looks like exactly is not very important, but here at the end, we have what is the end of the event itself. So it's a recovery part. And if we use the standard model that has no organic carbon burial in it, the black one, this very rapid recovery is missing. Whereas if we add organic carbon burial that is produced from a model using the restrictions or constraints that we have on productivity and oxygen, we actually get a very fast recovery. That's a bit too extreme. But when we limit it to a specific time interval in the PTM and a bit closer to what is realistic, we really get very nicely the overall pattern and the fast recovery. So this means that organic carbon burial also in the past was pretty important for drawing down CO2 at the end of rapid climate change. And the data that we have tells us that the climate change that occurred, which we consider somewhat of a worst case scenario equivalent for modern day climate change, was enough to cause this kind of large scale influence on atmospheric CO2, both through high productivity and decreased oxygen, both of which are things that are also happening in the modern ocean. And also this means that these are all processes that we definitely need to take into account when we're projecting through models usually what might happen in the future. Also sea level rise, because with higher sea levels, we might expect an expansion of marginal seas and shelf areas in the modern day as well, which could allow more organic carbon burial. And with that, I'd like to thank you for your attention. And I don't know if there's any questions now and otherwise later during the Q&A panel.
Thank you, Nina. Our next speaker is Hongjie Wang.
Everyone, this is Hongjie Wang, but I also respond to Hongjie. If you think Hongjie is too hard So today I will go over a few most recent paper on the eutrophication, hypoxia, and especially about nutrient management. And I will bring you some very exciting news about the nutrient management in the global ocean. And so next one. So make sure we are on the same page, understanding about eutrophication. So according to Marine Strategy Framework Directive, eutrophication is a process driven by enrichment of water by nutrients, N and T, and leading to increased growth, primary production, and the biomass of algae, and change in the balance of organisms and water quality degradation. So from this perspective, eutrophication is also known as the nutrient pollution. So because under eutrophication state, the excess of this algae will eventually sink to the bottom and got respired, and by using oxygen. So when the oxygen consumption rate is much higher than the oxygen supply rate from the mixing, the hypoxia is formed. And so next one. And so because of eutrophication, as Nina mentioned, since 1950, more than 500 sites had already reported hypoxia, and only less than 10% of them had hypoxia before 1950 because of eutrophication. And if you look carefully at this animation to describe the river nutrient loading to coastal regions, okay, since 1980 to 2050, this shared socioeconomic path fall. And so I highlighted a few case studies for you, area for you to look carefully. So general patterns from 1980 to 2015 each, the nutrient increase to the coastal region. And then around 2015, the nutrients start to decline and also continue to decline until 2050 with massive agricultural practice improvement. Okay. And so today I will use, again, with these three areas to represent, to share with you my understanding about the eutrophication state now in different, very typical coastal areas. Okay. So I call this as evolution of managed coastal eutrophication, and I will use the China's coastal area as the region to describe still at the present increasing eutrophication risk. So this map shows the Yellow Sea, East China Sea, and South China Sea, and here shows the nutrient loading to these three coastal regions for different, from different sector, different color represent the nutrient loading and to these three coastal regions. You can see clearly the nutrient loading to this coastal region increase. Okay, still increase for every single sector. Okay, because of the agricultural industry and urban streets, and also because of less governance and also enforcement. I have to say that. And so this, because of eutrophication, since 2008 in Yellow Sea and massive green tide outbreak happened in Yellow Sea. And this map, this figure took in 2008 when I was a student there in Qingdao. And and we, if not hundreds, if not thousands of students, go there to use our bare hands to pull as much as green tide from the Qingdao coastal region to prepare the Olympic Games. I still remember the desperation I was when I faced with like 10,000 meetings of football square green tide. It's exhausted work. But in 2021, the green tide area in Yellow Sea tripled. And this figure even shows clearly another impact for eutrophication. Chlorophyll concentration in Yellow Sea doubled for the past 15 years. And so in Changjiang River, Changjiang is one of the largest rivers in the global, and so the nutrient input there also increased. But good news is the increase really slowed down for the past 10 years. And this map shows the hypoxic zone area in Changjiang S3. So I want to highlight that when there's no data, it's no data. No means no hypoxia. Okay. And I didn't mean there's no action in China to try to slow down eutrophication, because you can see there's like the number of wastewater treatment in place to try to slow down eutrophication. But with that, the nutrient loading from the sewage still increased. But the good news is last year, the new program really tried to focus on the problem, and hopefully we can see some exciting results from this effort in a few years. And so next, I think there's no better ecosystem to represent us like a coastal region with a stagnated nutrient flux better than Gulf of Mexico. And so here's the nutrient loading to the Gulf of Mexico for the past 35 years-ish. And you can see the nutrient level, nutrient input, A and P, basically didn't change that much. And as a result, the hypoxic zone in Gulf of Mexico and kind of have us like, don't have a change at all. And this level of this, the hypoxic zone area is still way higher than the hypoxia task force goal, which was 5,000 square meter. And I had to say, it took less than 35 years to be this awesome, but it took 35 years, nothing for the Gulf of Mexico to make any significant change, okay? Next one, but I want to bring you some good news, okay? So for some coastal regions, they actually already passed this plateau period, and kind of nutrient input to the coastal region actually become to decline. And this, like a few case studies here from Baltic Sea and some US coastal regions, okay? And so this map shows here is the bathymetry of Baltic Sea. And so you can see this some areas more than 200 meters deep. And because of this very slow, limited water exchange with the North Sea, and the water residence time for the Baltic Sea is very long, over 30 years. And the good news is under the umbrella of this more than 50 years nutrient reduction action and the HELCOM, and so the nutrient loading to Baltic Sea started to decline after 2000, and for both A and P. Okay. The bad news is the ecosystem recovers slow, and we learned from yesterday, it takes some time for the ecosystem to respond to a perturbation. Even the perturbation is anthropocene and managed nutrient reduction, okay? And this map here shows the hypoxic area, anoxia area. Even though the hypoxic area declined, but anoxia area still increased with oxygen area. So mainly because the year-round stratification, and also the legacy effect from the benthic. And so, like Baltic Sea has such a big spatial difference, and there are some coastal embayment has a very exciting result after 50 years nutrient reduction. This is one highlighted by this star here. The DIN concentration, the average DIN concentration, and in this forge actually is almost like less than 50% of the original nutrient concentration. And so highlighted by this A, and this is the chlorophyll concentration also decline. You can see the trend. But the eelgrass only, even with this almost more than half nutrient reduction, the eelgrass only like at a moderate recovery. Again, from yesterday's lecture, we know it takes some time for the ecosystem to respond to the perturbation, okay? And we have to be patient. And now this is a wonderful review. Please read this paper if you are interested in the nutrient loading. And so they summarize all the nutrient trend in the North America. And so like you see nutrient loading in the Columbia River decline, also decline in the Great Lake and Delaware, Chesapeake Bay. And so again, I want to bring some good news. Okay, take the Tampa Bay, for example. You can see the nutrient loading to Tampa Bay significantly reduced for the past 30 years, and more than the current nutrient level is only like half of the previous condition. And the seagrass coverage also slowly increased after about 10 years of the reduction. You can even see the seagrass is a white index to show the recovery of the column kind of already passed the goal. Good, 10 years ago. And because I'm a limitation, I can't put too many case here, but you can trust me and go back to the paper to read more nutrient change, okay? But if I stop here, you may think the aggressive nutrient induction is a magic brush. You can solve all the problem and benefit every single person. But I want to put this case for you to think about this notion. So this is a lagoon in French, and he highlighted by the map here. And so this called the Sun Lagoon provides, it's a very, one of the largest Mediterranean coastal lagoon, support more than 10% of oyster production in France. So here shows the average DIP concentration in this lagoon, and especially you can see the sharp decline for DIP because of very aggressive nutrient induction and from the sewage and from the agriculture. And you can see the current DIP concentration is even less than 1/3 of the previous condition. So I want to challenge you to think about if we could compare the landing for the mussel with the black line, and the orange line is the oysters. You can see the landing declines sharply after nutrient induction. And the hypothesis is because this region changing to new region, shift to a new region because the nutrient reduction. And this is a maximum capacity under this new nutrient level. That means if with aggressive nutrient induction, you need expected to see some decline of some commercial fishing. And this is Narragansett Bay, and where I came from, Rhode Island, Narragansett Bay, and so the same thing also happened in Narragansett Bay. The green line here is the net primary productivity. In the Narragansett Bay, you can see clearly decline after nutrient reduction, and with this region shift, the one kind of a clam very popular to make clam chowder, if you ever try. And so that you can see the decline of the climbing landing in Narragansett Bay. If you get a chance to talk with the fishermen, they will tell you, I have never seen the bottom for the past 20 years since I get into this industry, but my clam is starving. The meat is thinner, and they can't get a good price because of less nutrients. They keep complaining, like maybe it's a bit too clean for them. But my point is, so I'm not kind of say, okay, now we need to like move away, like upside down of nutrient induction. My point is, I think we need to really think about the goal of the nutrient induction. Do we really want to make the water crystal clear, or we want to also benefit as much possible, as many people as possible? And so that's my question. What's the goal of the nutrient reduction? And earlier this year, this is a diagram Mark Brass and me draw, and basically we think in the future, especially for the coastal system which already went through very rigorous nutrient reduction, we may want to think about try to promote optimum nutrient loading, like to considering all the nutrient sources to the ecosystem, then to think and based on model, based on observation, to find a more optimum nutrient level to both benefit the water quality, look pretty, smell good, but also think the need for the fishermen, because a lot of people make a living of all the clam, oyster, carp, you name them, a lot of fishery, lobster, by the way. So from this perspective, so I don't want to call it's called a nutrient pollution, because if you use pollution, somehow you think you want to get rid of them because of pollution. So I think we may want to try to think about a better name to describe eutrophication. I want to lean away from. Okay, I will be there. Okay. So I will leave this page here. We can discuss. I summarized all the successful lessons we learned from the system that will already make a big progress about the nutrient reduction. And my take-home message is, we as a scientist, we need to make adjustment or recommendation as needed, no matter where the eutrophication state is. When the nutrient increase, we needed to try to quantify to get a criteria how to slow down, make suggestions. Then when the nutrient is so low, we may want to move to another direction. Hold on, slow down, slow down, slow down. We may want to look back, what's the goal? Okay, so that's my take-home message. Okay, with that, thank you for your attention. Thank you.
Our next speaker is Mona Norbisrath.
Yeah, thank you very much and welcome everybody. I'm very happy to be here today. Invitation today. So my name is Mona Norbisrath. I'm at Helmholtz-Zentrum Hereon and I work in the Institute of Carbon Cycle. Today I'm going to present you some aspects of metabolic alkalinity generation in adjacent zones of the North Sea. So we come back to the carbon a little bit more from this nutrient perspective. So as our session name indicates already, we are talking about marginal seas and in the Anthropocene. And when we talk about the Anthropocene, we are talking about the time after the industrialization in the 1850s. And during this time, we observed an increasing atmospheric carbon dioxide concentration. And in particular, in the last 16 years, we observed an increase of CO2 in the atmosphere of more than 100 ppm. And in order to tackle the global warming down to 1.5 degrees, as an aim of the Paris Climate Agreement of 2015, we have to reduce and also mitigate anthropogenic CO2. In addition to the increasing CO2 concentrations, also fertilizer and inputs of wastewater and nutrients impact in particular marginal seas. This nutrient and wastewater inputs are imported into the coastal ocean mainly by rivers, as you can see on this map here. And this is important to understand the happening and the biogeochemistry in this adjacent zones of the coastal ocean to also understand coastal carbon dynamics in the ocean itself. And due to its geographical location, coastal and marginal seas are the direct connection between the land and the open ocean and are strongly impacted by various natural impacts, such as an exchange with the air or the sediment, but also by anthropogenic influences, such as fisheries, transport, or the import of nutrients. In particular, the role of marginal seas as a CO2 exporter stays in focus, since they strongly take up CO2, which is then stored and exported into the deep ocean and stored there for longer timescales. Until now, 31% of the anthropogenic CO2 is stored in the ocean, which makes it the largest atmospheric carbon dioxide sink. Generally, the CO2 uptake in the shallow and marginal seas is either driven biologically by primary production or by the carbonate system and the driver total alkalinity. When we talk about the carbonate system in general, we mainly talk about dissolved inorganic carbon, and this constitutes of hydrogen carbonate, carbonate, and carbon dioxide. And both hydrogen carbonate and carbonate are the main substitutes of total alkalinity. Alkalinity is the acid buffering capacity, and it has the ability to neutralize acids such as hydrogen ions, and it's also defined as the excess of proton acceptors over proton donors. And the key role in this work is that the fact that alkalinity can be generated metabolically, and especially on shorter timescales. Some of this metabolic alkalinity generation processes, or the focus in this work, is either calcium carbonate dissolution, which result in our context of decreasing pH due to metabolic alkalinity activity, or by various processes of the redox system that generate alkalinity in various ratios. And the focus here is set on denitrification, which is the anaerobic bacterial reduction of nitrate into molecular nitrogen. And since this is an irreversible process, denitrification is a net alkalinity source. So the focus in my work is set on the coupling of carbon and nitrogen as a tool to better understand alkalinity regulation in adjacent zones of marginal seas, where I focus on a shallow adjacent zone of the North Sea, which is the Elbe estuary. And yeah, the nice thing here is that with the shallow water depth, we can combine and include all of the compartments like atmosphere, pelagial, and benthos to understand the biogeochemistry. So the Elbe estuary is a highly anthropogenically impacted adjacent zone of the North Sea, and our special focus is set here on the Hamburg port, which is Germany's largest seaport in the upper estuary. And in order to identify and quantify metabolic alkalinity generation in the Elbe estuary, we sampled along the estuary and divided the estuary in boxes for a box model calculation. And by using this box model approach, we estimated net metabolic alkalinity, DIC, and nitrate fluxes for each of the boxes as the closing term. And therefore, we assumed a steady state in terms of input and output of the box. We have calculated the baseline in accordance to salinity as a mixing between fresh and marine waters. And for this, box 1 in the upper box in the river and box 6 as the North Sea act as boundary conditions. And in order to complete oxygen and carbon dioxide, we also calculated their exchange with the atmosphere. So along the Elbe estuary, we observed alkalinity and DIC, and we observed a strong increase, in particular in the Hamburg port and in half of the estuary, with almost a doubling of alkalinity in the upper estuary. And when we plot this data, which are here above the Elbe stream kilometer, above salinity, we see that this strong increase mainly occurs in the freshwater 0 PSU region. And yeah, we normally would expect a linear mixing of alkalinity with marine waters in this linear increase, but we see this strong differ from the expected linear mixing. So we have and assume a highly metabolic activity in the upper estuary. This high metabolic activity is in the freshwater area is also visible in the strong oxygen uptake that we observed when the river enters the Hamburg port region. So we observed a strong oxygen decrease, and this decrease is also visible in our calculated air sea fluxes. So we see a strong oxygen uptake in box 2, which is the Hamburg port and river downstream. And beside this, we see a strong carbon dioxide production in the inner estuary, with both decreasing activity river downstream. So this strong oxygen uptake shows strong metabolic activity, oxygen consumption, led us to assume that we have, especially in summer and spring and summer, hypoxic and anoxic conditions in the benthic zones. So the strong connection of the atmosphere with the benthic zones are shown in this plot as well, and show the importance of combining all compartments in this approach. As a result from our box model approach, we got net dissolved budgets per box, and we identified box 2, which is the Hamburg port, as the one with the strongest metabolic activity. And when we look at the DIC bar in gray and see its CO2 SC flux in light gray, we see that we have, compared to the produced DIC, a relatively small outgassing. But when we now compare this both chart in the downstream boxes, we see that the production of CO2 decreases river downstream, but the SC contribution stays similar, the same. So we assume that we have here a transported signal of CO2 produced from the port downstream the river, and the equilibration time is not enough to equilibrate with the atmosphere. When we now look at the alkalinity generated in the estuary, we see, and when we combine all of them, we got to an overall metabolic load of metabolically generated alkalinity of 14% from the entire Elbe estuary. And when we use this and combine this with particulate matter properties, we attributed that main part of the alkalinity was produced or has the source in particulate inorganic carbon. And so we assume that most of the generated alkalinity could be generated by calcium carbonate dissolution. And we assume that the remaining 10% of the metabolic alkalinity in the estuary then was generated by other anaerobic processes. However, with this coupling of carbon and nitrogen, we were not able to further and clearly attribute denitrification as source for the remaining 10%, since the nitrogen cycle has too many processes that affect and influences this, such as nitrification. But we can clearly say that main part of the alkalinity in the Elbe estuary was or has calcium carbonate dissolution. When we now take the results of the box model approach in order to investigate the effect of anthropogenic changes on the coastal ocean, we used the metabolic fluxes from the box model, and we used also a biogeochemical model approach of the 3D EcoHem model in order to see if a change in metabolic loads would also change the CO2 uptake in the North Sea. And therefore, we used like the normal 100% reference run of all alkalinity generated and transported down the river. And we used this 100% and reduced it by the 14% of the metabolically generated amount, 86%. And we also decreased the alkalinity load down to 50%, but also increased it up to 150 to see a comparison if we have a change when we have, or when we would have a higher alkalinity production in the upper estuary. And by doing so, a result of this model was absolute RC fluxes of carbon into the ocean at different distances at sea. So we have 100, 150, and 200 kilometer distance from the coast. And when we now look in one distance, we can see that with a higher alkalinity load, we also have a higher carbon uptake in the ocean in every distance. But when we now look between the distances, we can see that a lower alkalinity load, or with a lower alkalinity load, the highest uptake of carbon still occurs in the open ocean. And the continental shelf pump shows here a stronger, or has a stronger impact and has a stronger effect than the uptake in the coastal ocean. However, when we now increase the alkalinity load strongly, like we did with this 150% increase, this changed, and we now have a higher carbon uptake in the coastal ocean. So with stronger alkalinity production in the coastal or adjacent zones of the North Sea would lead to a stronger carbon uptake in the coastal ocean. So coastal oceans would act as a stronger carbon dioxide sink under enhanced alkalinity generation in adjacent zones of the North Sea. And as concluding remarks, yeah, I would say that the inclusion of biogeochemistry in adjacent zones of marginal seas is important and shows the important role in the coastal carbon dynamics. So we should include also the stuff going on in adjacent zones, in rivers and estuaries, when we talk about coastal carbon uptake in coastal oceans. We saw that an alkalinity control shows an effect on the CO2 uptake, which doesn't mean that I suggest to put a lot of nutrients into the inner estuaries and see what happens then. So we don't know the effect on other processes yet, so this just needs more work. Furthermore, I suggest that an inclusive approach, like to combine the atmosphere, the pelagial, and also the benthos is needed and required to get a whole picture. And furthermore, the coupling of carbon with other nutrients such as nitrates is a useful tool to better understand metabolic alkalinity regulation in adjacent zones of the marginal seas, but also in the marginal seas itself. And with this, I thank my co-authors and also our funding agencies and our Make Our Planet Great Again project for funding this research. Yeah. I'm also looking for a postdoc position, so if you have one, please let me know.
Brenda's already here. Our next speaker is Brenda Carter.
So I made a mistake. I was working from an old email, and I was planning around a 20-minute talk. This is a 12-minute talk, so we're going to flip through some slides really, really quickly. In the session organizer's defense, I checked. Oh, I have 15 minutes. Wonderful. They made that very clear in subsequent emails that I missed. So sorry, guys. No, I'll just go quick. Okay, thanks. Okay, so my name is Brendan Carter. I work for the University of Washington, but I work at the NOAA Pacific Marine Environmental Laboratory. I have kind of a sweeping talk title, which is Quantifying the Intended and Unintended Impacts, Human Impacts on the Carbon Cycle of Marginal Seas. When I say unintended, really what I just mean is anthropogenic carbon storage in the ocean. That's ongoing. It's happening whether we like it or not. When I say intended, I'm really just talking about marine carbon dioxide removal. Another caveat, I'm really intimidated to be in a section on marginal seas because for most of my career, I have a very global focus, and my interaction with marginal seas has been kind of whiting them out at the last minute in the publication because we don't actually know what's going on there. But I feel like that's probably a useful perspective for this session because I can kind of maybe share some perspective on why I keep having to do that. Okay, so this is the whole talk. It's kind of divided into 3 parts. The first was intended to be about the challenges that we face when evaluating human impact. I'm going to go really fast through that. Anthropogenic carbon estimation in marginal seas, I'll slightly slow down there. And then this is the really interesting question that was posed when we were kind of thinking about this talk, which is, would marginal seas make for good MCDR testbeds? I put the morals here, so anyone who needs to just take a picture and go get coffee, you can do that. Okay, so this slide was just meant to be why it's hard to quantify human impacts. And it's really because what we're talking about is a very abstract thing. It's the difference between our world, kind of represented by that middle Earth, and some other Earth that doesn't actually exist without humans, or alternatively, some other Earth in which we have done some aggressive marine carbon dioxide removal. We can't put sensors on these other planets, so we are stuck with trying to make inferences about how different these worlds are. We can do this maybe with time series, but anyone who's worked with time series knows that there's a whole lot of natural variability, and the anthropogenic signals that we're looking for are often very small relative to that natural variability. I'd planned to explain all these, but I'm not going to. So we have ways of mitigating that natural variability. There's all kinds of clever things that we can do with well-resolved time series in the surface ocean, deseasonalization, so on and so forth. In the interior ocean, we rely upon co-located measurements of a lot of different properties like temperature, salinity, oxygen. We can take that co-located information and start to look at how carbon is varying in relation to those other parameters. And the processes that lead to natural variability in the interior ocean also lead to natural variability in these co-located measurements. At the end of the day, we can pull out something that looks a lot more like what we'd expect to see from anthropogenic carbon, with accumulation in the surface ocean, deeper accumulation in places where you have intermediate water formation, and less accumulation in places where we have upwelling of deeper, older waters. All of those methods, however, require a lot of data in order to really make them work. And so let's kind of focus on the marginal sea places. This is the GLODAP data product. This is not the only data product, but it's one of the places where those of us who have a global focus on carbon often begin and end our days. So let's look at the marginal seas. You can see this is the first 10 years of that data product. We've got some coverage in some of the marginal seas, but not a lot. Fast forward 10 years, we're doing a little bit better. We're still pretty barren in a lot of them. Another 10 years, and Mediterranean now has a great set of cruises, which I'm hoping Marta will talk about at least a little bit. But we're still kind of suffering. Okay, there are other data products. This is a plug for the GOMEC4 data set, which I think is going to go into the CODAP data product. CODAP is really focused on North America. But my point here is that there are data sets in all of these regions that just aren't in GLODAP, and we need to get them there. So if you have contacts in these communities that are working on these things, get them into data products, make sure that those kind of get out there, and then the global community will start working with them. I had a series of slides here on CFCs, transient tracer observations. This is another way that you can get at carbon information even without measurements of carbon. You can start to quantify histories of contact between the atmosphere and the ocean and use that to infer anthropogenic carbon. I'm going to kind of skip through these though. The same point was going to be made here. We're missing a lot of CFC, CFC and SF6 information in marginal seas. This is CFC 11, and this is SF6. SF6 is really important for modern estimates of this kind of stuff because the CFCs have been banned since 1990s, and now we're kind of relying upon new transient tracers. Okay, this is a slide from a kind of recent global synthesis, just showing sort of the state of things. The bottom panel shows where things stood with global carbon inventory estimation as of, I think this is the Sabine et al. Analysis. And then the top part is everything that they've kind of added since using these global analyses. And the point I wanted to make here is we're still blocking out most of the marginal seas. So this is kind of an ongoing problem. This is a summary statement for this first part. And I would say anthropogenic carbon in marginal seas could use more attention. Here are three things that I think could really help with that. Just more routine monitoring, more transient tracer observations, and most importantly for this crowd probably is better marginal sea data integration into global or at least regional data products that will then feed into the global data products. I also wanted to mention that biogeochemical Argo could change the way we do a lot of this kind of science. We don't yet know how that's going to shake out, but we're excited about it. And thanks to folks like Emily, we do have some floats in some of these marginal seas, and there's quite a few in the Mediterranean. Okay, so now moving on to the marine carbon dioxide removal portion. A lot of this is focused on work that we released recently. Hongjie, who just gave a talk, led that paper. This is kind of the rest of the team that was working on it. This was a simulation of the release of a whole bunch of alkalinity. Let's see, do I have a pointer? Okay, there's a red dot on that map up there on the far left. Simulation of the release of a whole bunch of alkalinity in Unimac Pass, Alaska, and then we were tracking the kind of chemical plume that was coming off of that and going across the Bering Sea. The point I wanted to make here is the same point I was making earlier, which is that the signal-to-noise ratio is tiny. We're talking about, this is at the M5 moorings, one of the black dots near the top, 3/5 of a micromole per kilogram of total alkalinity, and this is despite it being a rather huge alkalinity release. 1/2 a micromole per kilogram of dissolved inorganic carbon once you're that far out. You compare that to the natural variability, and its natural variability is something like 80 to 300 times as large. So it's going to be really tough to pull these signals out of natural variability on the spatial scales that we're talking about here, at least. Once again, there are ways of dealing with MCDR, and this is kind of a shameless plug for some of the research that we're doing in our team. This is an algorithm estimate of alkalinity. There are algorithms that you can use to estimate just about anything in the global ocean now. Over x-axis is kind of measured alkalinity. This is the GLODAP data product. The y-axis is estimated alkalinity using these algorithms. And just the point that we wanted to make here is that generally speaking, we have pretty good ability to estimate one property from other properties as long as you have the right set of measurements. These can be used for MCDR. You can take your estimate, say this is what we would expect it to be, and then kind of compare it to the measurement that you have of alkalinity, and then say, okay, maybe this difference here can be attributed to alkalinity or to MCDR. Same could be done for direct ocean capture if you're looking at dissolved inorganic carbon. A lot of these claims I'm making, I would argue, are generalizable across MCDR approaches. But caveat one is that the signal has to be big enough. So you still have to have a kind of confined signal or a large signal. Caveat two, specific to marginal Cs, is that when you try to apply these algorithms in places where you don't have any training data, you generally find much larger uncertainties, much larger errors. And so kind of one of the morals of our past work in algorithm development is that you really need to do at least validate the algorithm in the place that you intend to use it, and better yet, develop an algorithm that is specific to that region. And that definitely applies to marginal Cs because they are usually unique in biogeochemical perspective. Call out for Larissa Diaz, who is somewhere here, I believe. There she is. She's leading our kind of next generation algorithm development work. Okay, so for marine CDR, I feel like to answer the question of whether or not marginal Cs could be good test beds, you really need to think about what questions are we trying to answer in MCDR right now. And I would argue those questions fall into two categories. First category is related to co-benefits, disbenefits. That's kind of jargon, but it's just asking, what are the local scale impacts? Could I get this permitted? Is this going to be safe? Is this going to cause a hazmat site where I'm doing this thing? Hazmat, super fun, that's what I meant. Are there ways that you can monetize this by farming kelp, so on and so forth? Those are all very local questions. By contrast, the second set of questions relate to additionality. It's, can I make money selling carbon credits on this? Does this actually remove carbon from the atmosphere in net? And if so, for how long is that carbon out of the atmosphere? Those, I would argue, are very global questions. But maybe marginal Cs are kind of the Goldilocks spatial scale where they can sort of address both sets of questions. That was my hope in the study. So let's kind of explore whether or not that's true. I'm mostly going to talk, these are three different pH thresholds, and what's being shown on the plot on the left is the area of our Bering Sea kind of region that meets that threshold of perturbation. So this is to some degree how much is ocean acidification mitigated by releasing all this alkalinity. Put these three little blobs here so that you can kind of get a sense for the relative area. The blue is basically unmeasurable. Even the best pH measurements out there could not measure that plume. The red is measurable. The best pH measurements could measure that, but there's no way you're going to pull out a pH signal of 0.002 from all of that natural variability. Yellow, which you probably can't see on the map on the right, is this tiny little dot represented by this kind of wiggle at the bottom. And again, that's on a that's on a log scale, so that's a much, much smaller area. That is probably detectable amid natural variability, but you can see that that line is not continuous. It keeps dropping out, and what's happening there is that there is not even a single grid cell in our model that is meeting that threshold at that time. So that suggests that maybe this model has too broad of a spatial scale for these kinds of questions. This is the slide on kind of follow-on research that we're doing. We're setting up a test facility at the Pacific Northwest National Laboratory in Sequim. Zoomed in all the way on the right, if you kind of move to the middle, this is the Sequim Bay. You move to the left, that's the Strait of Juan de Fuca, and you can see how small the Sequim Bay is. These are the regional scales that we're doing our modeling on for our subsequent follow-on experiments. Much, much finer resolution than you would have in a marginal C necessarily. Also, this little triangle here, that's one grid cell, and we have some modeling work that is even going below that spatial scale. So we have subgrid scale modeling, really to try to plumb the local scale impacts. Okay, global scale impacts. This was our attempt at trying to do the global scale impacts with this marginal C model. What we were able to show is that the carbon dioxide removal potential expressed here in dissolved inorganic carbon units, we were able to show that most of it is realized, as represented by this plume of unrealized carbon dioxide removal potential, mostly going away. So most of it is realized within our domain. That was good. That suggested that one of our biggest concerns, which was how quickly does the atmosphere re-equilibrate with the alkalinity kind of perturbation that we released, that was happening within our model domain. It was happening fast enough. We could use this model to answer that question. But it still doesn't answer additionality. For additionality, you need to know not just how long does it take to equilibrate, but you need to know what is the ultimate fate of that water. It's leaving our model domain with a different carbonate system than it would have had if we hadn't released this alkalinity. That means it's going to respond differently to air-sea gas exchange. It's going to respond differently to biogeochemical cycling. And we really need to know the full global fate of that. So I would argue for additionality, you really need to be looking at global modeling. This is just one example. There are many, many papers out there that are using these kinds of global models. So summary statement, are marginal seas good MCDR test beds? Are they the Goldilocks of spatial scales? I would argue that they are unfortunately an unhappy medium where they can kind of reach out and touch both ends of the questions that we care about, but they can't truly answer them. So I think we'd probably be better off making sure that the spatial scales that we're doing for our MCDR are really targeted for the exact questions that we are most concerned with answering. And hey, 13 and a half minutes. These are my funders. Thank you very much for keeping me going. And these are the people who employ me. Thanks.
Our next speaker is Sophie Gill.
Fantastic. Right. Okay. So hi, everyone. I'm really excited to be speaking to you today about the Black Sea as an analogue for ocean alkalinity enhancement. I just want to say thank you so much to the organisers for inviting me to speak today, because I was super excited when they reached out about giving a talk, because I actually attended OCB for the first time back in 2019 as a graduate student, and it's one of the best conferences I've ever attended. And back then I gave a lightning talk about some of my coccolithophore culturing experiments, so it's just like really amazing to kind of reflect back on that and yeah, be invited to speak today. I now work at Isometric as a carbon removal scientist, where we're building a carbon removal verification and science platform, and it's super exciting to see so much marine CDR research being discussed and being presented at this conference. So today I'm going to be talking about a few different things. First of all, I'll introduce the science behind ocean alkalinity enhancement as a carbon removal technique, so everyone's on the same page with that. And then I'll talk a bit about a project that I was really fortunate to be involved in during my PhD, headed up by Leonard Bach, where we reviewed the state of research into biological responses to OAE, and there were some really interesting reflections about the Black Sea from that. And then I'll briefly touch on some culturing experiments I did during my PhD, which weren't in the Black Sea, so I hope everybody forgives me for straying slightly from the marginal seas, but they were at fairly similar carbonate chemistry conditions to the Black Sea. And so I thought it'd be really interesting to look at that data here. So first of all, let's dive into the science behind OAE. So there's a multitude of different definitions of ways of thinking about alkalinity as a concept in the oceanographic literature, which I'm sure everybody's familiar with, but I've kind of landed on my favorite way of thinking about it over the course of the last few years, which is starting from the principle that in the oceans, we have an excess of conservative cations compared to conservative anions. And actually on this scale of this diagram, this looks very small, but and I guess on the scale of the global ocean it is, but this conservative just means that concentration can be assumed to be constant throughout the global ocean because they're so abundant. But this excess can be described chemically by the explicit conservative expression for alkalinity. And the sum difference between the conservative cations and conservative anions can be described by the carbonate system components that can change their speciation in response to perturbations in ocean chemistry. And the Geryn plot here can be used to illustrate how external perturbations of pH and alkalinity can shift the carbonate system between its three major components. So we have aqueous CO2, bicarbonate ion, and also carbonate ion. And alkalinity can be increased by adding conservative cations to the surface ocean, for example, through weathering processes on land, because this increases the difference between the conservative cations and anions in the surface ocean, and this causes the carbonate buffer system to shift its speciation towards this more negatively charged end of the spectrum to make up for that extra charge imbalance. And this means increased levels of bicarbonate and carbonate ion and decreased aqueous CO2. And due to Henry's law, decreased aqueous CO2 promotes dissolution of atmospheric CO2 in the surface ocean, therefore drawing down atmospheric CO2. And this acts, we all know this acts as a negative feedback on carbon emissions within the carbon cycle and prevents potentially runaway greenhouse climate effects. So we can see that if we were able to accelerate or simulate this natural process to add extra alkalinity to our oceans, in theory, this could be one way to actively draw carbon dioxide out of the atmosphere, and hence where OAE as a potential carbon removal technique comes from. So during my PhD, I was invited to be involved in a project headed up by Leonard Bach, where we wrote a review paper on the current status of pelagic ecosystem, the current status of research into the potential response of pelagic ecosystems to OAE. I also worked with my supervisor, Ros Rickaby, as well as Sarah Gore and Phil Renforth on this paper. And we were particularly interested in looking to the geological record, as well as natural analogues in the modern day environment, to examine how pelagic ecosystems might respond to OAE. And in the paper, we presented two different scenarios for OAE, determined using carbonate chemistry speciation modeling in CO2-CIS. The first is air equilibrated OAE, which describes a situation in which the surface ocean and atmosphere are equilibrated with respect to PCO2. So therefore, PCO2 will remain constant with increasing alkalinity, but pH and saturation state of calcite will increase, and you can see pH and calcite are on these. So pH on this graph, saturation state of calcite on this graph. It's important to consider, though, that an air equilibrated scenario of OAE might not occur on the initial addition of alkalinity to the surface ocean, because reaching this equilibrium could take several years. So when alkalinity is initially added to the surface ocean, it might be added too rapidly for air-sea gas exchange to keep pace, and we refer to that as non-equilibrated OAE. And that means that more extreme carbonate chemistry perturbations would occur, leading to potential hotspots of alkalinity addition and locally severely decreased PCO2, in addition to increased pH and saturation state of calcite. So alkalinity addition dynamics after air-sea gas exchanges occurred might result in different carbonate chemistry perturbations compared to non-equilibrated conditions. And this graph on the right here showed some predictions we made about relative calcification of Gephyrochapsa oceanica, which we saw that under air equilibrated OAE, we might expect increased calcification, and under non-equilibrated OAE, we might see decrease. And Gephyrochapsa oceanica is a coccolithophore, by the way, didn't actually mention that point. So this was, some calcification sensitivity of Gephyrochapsa determined in experiments and fitted to Leonard's coccolithophore calcification model. And what I'd like to sort of draw your attention to here is just this box on all the diagrams showing where the Black Sea sits on this PCO2 alkalinity space. And the Black Sea is a suitable analogue for a highly perturbed OAE marginal sea, as it has a surface alkalinity of about 3,300 micromoles per kilogram already today on average. And the major planktonic processes in the Black Sea are similar to other oceans, but there's like one particular thing that's quite interesting about the Black Sea, which is that it harbors some of the most extensive blooms of calcifying phytoplankton, known as coccolithophores, and thick layers of calcareous ooze in the sediments. And you can see these blooms here in this really nice satellite image from the NASA Earth Observatory. And these blooms are likely promoted by the favorable carbonate chemistry conditions, since coccolithophores are largely absent in the Baltic Sea, which is a lower alkalinity marginal sea, but in other respects quite comparable to the Black Sea. And when we were looking into this at a high level, Leonard suggested that he could hypothesize like an overarching framework to encompass how the material used for alkalinity enhancement would not just affect specific subgroups of phytoplankton, like calcifiers, but could have impacts on the relative success of other major phytoplankton functional groups, because they might have contrasting optima with respect to carbonate saturation or pH, as we'd kind of looked at. For example, total alkalinity will increase during an OAE scenario, and calcium might be added to the surface ocean to do that. And we predicted that these carbonate chemistry conditions would be favorable for calcifiers. So there's a diagram of coccolithophore here. And Leonard suggested that we conceptualize the modern day ocean as a blue ocean in ecological equilibrium between the major phytoplankton functional groups of calcifiers like coccolithophores, silicifiers like diatoms, and nitrogen fixes like cyanobacteria. And in an OAE scenario, the increased relative success of calcifiers could shift the ocean to a whiter ocean. But by contrast, if silicate materials, for example, olivine, were used to enhance alkalinity, increasing total alkalinity would be accompanied by a rise in dissolved silica and trace metal enrichment like iron or nickel. And silicifiers could likely benefit from an increase in dissolved silica, and nitrogen fixes could be more successful due to these trace metals being added. And these responses would result in overall increased primary productivity until macronutrients were limiting, and calcifiers could be outcompeted. And this could be thought of as a shift towards a more green ocean. And testing this hypothesis obviously requires looking at a full suite of like mesocosmic experience modeling, but we also thought this could be realized by studying natural analogues like the Black Sea as an example for a high alkalinity marginal sea. So how can the Black Sea give us an insight into how plankton could respond in other areas of our oceans? And are we likely to see blooms of coccolithophores as a result of OAE, for example? And a particularly interesting aspect in the context of community shifts is this competition between calcifiers and silicifiers. And Humboldt et al. In 1997 actually provide a really interesting case study where this effect of community shifts between calcifiers and silicifiers driven by external perturbations may actually have been observed. So the River Danube contributes about 70% of the river inputs into the Black Sea, but was dammed in the early 1970s. And the damming reduced dissolved silica inputs into the Black Sea and promoted coccolithophores at the expense of diatoms. And a prerequisite for the success of calcifiers under OAE would be that growth of the usually more competitive silicifiers is kept in check by dissolved silica limitation. So under silicate-based OAE, this limitation might be relieved so that the fertilization of calcifiers through alkalinity enhancement could be overcompensated by dissolved silica fertilization on silicifiers. So that's like the reverse case of what happened in this case study. And it's just interesting to address this issue because it's very important because the balance between calcification and silicification has like a big influence on the efficiency of OAE. So for example, in theory, if we think about it, if dissolved silica inputs reduce net calcification and the subsequent export of calcium carbonate to the deep ocean by about 10%, if you then accounted for that, if you think about that in a mole per mole basis, then about 20% more alkalinity would remain in the surface ocean to absorb atmospheric CO2 because of the stoichiometry of 2 moles of CO2 drawn down in the surface ocean for each mole of conservative calcium ion added to the ocean. And that could enhance the CO2 uptake of the oceans and increase the effectiveness of OAE. So we can see that this example from Humboldt et al. It really nicely illustrates how we could use the Black Sea as an analog in practice for OAE. But and there are situations that have happened there that provide a kind of a natural test for seeing these perturbations, which we hypothesized about in the white versus green ocean scenarios. And just to touch briefly on this before the end of the talk, so I've actually also worked on laboratory culturing experiments with coccolithophores, not from the Black Sea, but from other areas of the global ocean to see if the hypothesis of higher likelihood of blooms being more likely under calcium-based OAE is true. And this work is currently unpublished, but I'll hopefully be submitting it soon. We went up to alkalinities between 3,000 to 4,000 micromoles per kilogram, which are on the high end, like the Black Sea, which are in all likelihood probably far above what's likely during alkalinity addition deployments by carbon removal suppliers working in the space right now. But this acts as an examination of the upper envelope of what we might expect and gives us, that kind of upper range on what might happen. And I measured calcification and growth of 2 different M-member species of coccolithophores, Emiliania huxleyi, which is a small, fast-growing coccolithophore around 2 micron diameter, and Coccolithus sparudii, which is a much larger coccolithophore to these high enhanced alkalinity conditions. And results showed that for E. hux, which is the primary bloom-forming coccolithophore in our surface oceans globally, as well as actually in the Black Sea, calcification per cell did not increase under air-equilibrated OAE conditions, but growth was fertilized by increased availability of bicarbonate ion under the enhanced alkalinity conditions, which overall means that we might expect higher population levels of E. hux under OAE, which would mean more calcification at an ocean system level because of a high number of coccolithophores doing calcification. But of course, this was only a lab experiment, so we want to verify this with some sort of modeling work as a complement. But for Coccolithus sparudii, their growth was unaffected by being placed in OAE conditions relative to control, which is likely because they come from a different genus than EHUCs, and this means they're less able to utilize alternative carbon species for metabolic processes like growth than EHUCs. And we likely think that high pH and low CO2 in the OAE conditions drove decreased calcification per cell in brow radii, and this has been shown in some similar experiments by Gerald Langer and others in the past. So we did see an indication from our laboratory experiments that if alkalinity was pushed up to the high levels we observe in the Black Sea, we might observe the same coccolithable blooms dominated by EHUCs that we observe in this highly perturbed marginal sea today. And just to note as well, from an MRV perspective, so that's monitoring, recording, and verification, because calcification releases CO2 as a byproduct, it's really important to constrain weather calcification from marine organisms like foraminifera and coccolithophores might result in some re-release of the CO2 that you're originally trying to draw down through alkalinity enhancement. And I guess just kind of take-home message would be, I feel like what Brenda's shown us shows that maybe doing experiments in these marginal seas might not be the best route forward for testing marine carbon dioxide in some instances, but I would probably urge that there are some instances like the Black Sea where we can learn something about that is kind of almost like a natural experiment that's already happened, because these marginalized seas that are away from the influence of some of the larger oceans, currents, etc., that might influence things, and we can maybe learn a lot from these kind of higher alkalinity, marginalized seas. I just want to acknowledge thanks to the following organisations. I worked at the University of Oxford in Ross Rickaby's lab, Oxford Ocean Bug, during my PhD, where the work I've presented today was completed. And I worked as part of the UK Greenhouse Gas Removal Research Programme funded by NERC. And Isometric is my current affiliation. And a shameless plug that we're hiring scientists to work on MRV of marine carbon dioxide removal. So if you're interested, get in touch. Jing from Isometric is also in person at OCB and giving a talk this afternoon. So definitely go and find her if you'd like to find out more. And thanks for listening. I'm looking forward to discussing more in the Q&A.
Okay, so we're going to move into our Q&A session. So Brendan, Mona, Hongjie, if you wouldn't mind coming up to the front. Thank you so much, Sophie, for that beautiful talk. And Nina, thanks for coming back on camera. So we have about 10-ish minutes here to take questions before our break.
So we've heard a little bit from all of our speakers about how ecosystem responses to these perturbations, all of the perturbations that we've talked about, might be slow. So in a context where we're also starting to discuss geoengineering, climate interventions, things like CDR, how do you think we can balance the slowness of that response with also trying to be as environmentally responsible as we can be and sort of limit some of those impacts? Curious about the panel's thoughts.
So I think
the mic.
Okay. Yeah. Okay. So just that's a wonderful question. I don't have a good answer, but I can try to use nutrient management as an analog to maybe can get some inspiration for the OAE on the way. Like as you said, like for the Tampa Bay, take Tampa Bay for example, even though they have such aggressive nutrient reduction, it takes more than 10 years to see the impact. Same with Chesapeake Bay, more than 10 years to see this part, and take more than 20 years for the Baltic Sea to see the impact. So that means prepare for 10 years funding before see the result. That's how I can think of now.
I don't think we have a lot to add to that. Thanks.
Sophie and Nina, do you have any comments on that before we move on to the next question?
Yeah, absolutely. I mean, it's a great question. I would probably point to the role of really rigorous MRV in ensuring that we do the best job possible in doing responsible carbon dioxide removal. Yeah, getting that right is going to be super important. And probably just to bring it back to the marginal seas as well, I do think there's some opportunity to learn from both marginal sea situations, but also possibly pointing to examples in the geological record where we could learn a lot about what might happen at different alkalinity conditions, for example, in the case of OAE. And I think that's also really important to reflect on, particularly for the academic community, and we can then take that into practice as well.
Great, thank you. I have an online question for Mona from Alec Wong. Do you have any PIC data into and out of the estuary for your box? Is it possible to construct a PIC model to show that total alkalinity generated in the estuary is indeed from PIC dissolution?
Yeah, so we had the PIC values from the whole estuary and also the entering stuff. And this is just an upper limit. So we wouldn't say that all of this alkalinity is totally generated by it, but we think that out of this stuff of PIC that is available, the 90% of alkalinity generation could be generated by the PIC available in the estuary.
Along those same lines, I'm wondering if you look at the carbon isotopic distribution of the alkalinity in these transition zones, and in particular, differentiating alkalinity produced by sulfate reduction in these shallow environments, which can, a benthic flux of alkalinity from the carbon, organic carbon pool versus the PIC pool. Do you ever do those kinds of things?
No, we haven't done this in this estuary, yeah. But yeah, of course, this is also, like I mentioned before, that the inclusion of other parameters important. We just started with originally only with the nitrogen part and then extended to this calcium carbonate dissolution thing. So of course, other data and parameter needs to be included to get a whole picture of how metabolic alkalinity is really generated in there. So this is just the first step, also to show what could be the sources and how big is the amount of produced alkalinity in the adjacent zones of the North Sea and how is the impact of them and how could they be probably enhanced and what would it mean for coastal carbon dynamics in the ocean.
Now, I still have this instrument, so I ask a question of Hongjie. How do you estimate the groundwater inputs? When you showed the coastal inputs of nutrients, how are you doing that, particularly in the Asian system?
So in Asian, let me think. I think the project I show you, I didn't look at their methodology. I assume they do use some radioactive radar, maybe try to quantify the groundwater discharge, and then. But I can speak for Narragansett Bay. This group of scientists in GSO, they quantify the groundwater nutrient discharge in one of embayment, only one. There's so many embayment around that region, but frankly, there's no solid conclusion how much groundwater discharge is contributing to the total Narragansett Bay nutrient budget. That's our goal. We want to get a clear budget before we set up the optimal nutrient level. Yeah, thank you.
I think that's a really important thing because in your patterns, it's the same person, that input dominated many of the magnitudes of your flux patterns. So critical.
Thank you. So like the Narragansett Bay, even though it's very small on the map, for the people living there, it's still big enough. So for kind of offshore area, water is very clean, and for the embayment, you have hypoxia. Occasionally have hypoxia. So it's very challenging to satisfy all people's need. Like some people, okay. So yeah, I can stop here.
I have a question for Hongjie.
Okay.
So actually, there's two questions. It just mentioned about the river nutrient export in the Changjiang River and the Mississippi River. So I have a question about, does this increase in river nutrient export, that is the global phenomena or specific to those large river system? And another question is, can we derive a conclusion that the net community production in those river systems on the shelf, they are increasing or not?
So for the nutrient budget part, overall, the kind of the pattern is, if you take Changjiang Estuary, for example, the large river estuary, because the reason it's large because it runs so many basins, so many areas, and when it's big enough, it runs through agriculture zone, and for that, it's very hard to regulate the nutrient discharge from agriculture zone. And for the EPA, for example, they have some exceptional requirement and criteria for the agriculture discharge, so they can set up a very high standard like regulation for the wastewater treatment, but they have some exceptional criteria for the agriculture runoff. The same with China, and I really didn't find a clear kind of the regulation about the agriculture runoff. So I think this kind of large river have a very similar problem with agriculture make big contribution for the total nutrient budget. And so if I have to conclude, maybe it's kind of overall trend, the nutrient loading from a large river still increase for a lot of large river estuary. For the NTP, I don't have a good answer for the net community productivity in the coastal region. But theoretically, in principle, when you have more nutrient, you may trigger a bigger plume. But I don't have a good, big picture of how they change over for the past 2 decades. That answers.
Oh, it got answered online. Thank you.
Sophie took care of it.
Hi, I have a question for Mona. And as your figure illustrates, most of the alkalinity addition and DIC addition happen at the very upstream of the river. And if it's carbonate dissolution, do you have an explanation for why it is mostly upstream? And also, in addition to that, do you have an estimate about how, what's the speed of the addition? Like how fast is the carbonate dissoluting into the river? Like within 24 hours, is it a long time span? Thank you.
Yeah, so first question, the most productive area was the Hamburg port, and we assume this as due to the high metabolic activity and the pH decrease. So we have a very strong pH decrease in this region, leading to calcium carbonate dissolution. But as I mentioned, we only use this as an upper approach or as an upper limit. We don't say that all of this really happens there. But when we use this data, this could lead to an alkalinity generation of this 19% metabolic generated alkalinity.
Final question.
I have a question for Nina. This is Ika Zhang, by the way. So I heard carbon isotope was mentioned, and that prompted this question. You showed that there's a lot of organic carbon buried on the shelf during PETM, and you mentioned, of course, there's like anoxia, there's probably like enhanced nutrient from weathering hydrological cycle. But is there any like isotopes or like lipid biomarkers or something done on those organics to distinguish between whether they have like a marine origin or a terrestrial origin? And I ask because, this is also, yesterday people mentioned about like the Q10 pattern and temperature dependent metabolic rate, which is something that we have seen in terms of like a bacterial degradation of organic matter. So I wonder, of course, there's a balance between all of these factors, but by knowing like where do they come from, it probably helps. Any kind of work like that has been done?
Hey, yeah, thanks for your question. Definitely, it depends a bit on the location and very much on the condition of the sediments and the organic carbon, whether they can do this. For the location that I showed with the nitrogen isotopes, for example, in that same paper, they have looked at, I think it was the nitrogen isotopes itself versus the carbon isotopes to see that there is very much a mix. Whereas before the event, for example, it was all marine. During the event, we have a lot more terrestrial material coming in. And this is a pattern they see in a lot of sites just because of enhanced hydrological cycling. There's a lot of sediments coming in from land and a lot of organic matter. It's still very much an ongoing research topic about what the relative contribution was on a global scale and how much of the organic carbon that we find is actually newly produced and therefore actually a sink for carbon, or whether it's weathered organic carbon from land, which could actually be a source of CO2 to the atmosphere. But it's definitely something that's being done a lot. And it's very much a question also for especially the marginal areas, let's say.
Gotcha, thanks.
I think we're wrapping up, yeah? Yeah, okay. We'll have another big panel discussion where we can ask more questions, but one more round of applause for our speakers. And we'll go to break and reconvene at 11.
All right, so let's get started. Welcome back to the Marginal C session again. And so earlier this morning, so we were mostly focusing on the processes. Now we're going to go to geography, so different places. I would like to make a request. If you're sitting in your area of your favorite Marginal C, if your speaker comes on the stage, make a wave. But don't drop your computer, okay? So the next speaker is Marta Alvarez from Spain, and she'll be talking about the cycle in the Mediterranean Sea.
Well, thank you so much. Now I feel more relaxed about this talk because I'm really nervous. So first, I want to say thank you for the organizers, Xinping and Emily, for inviting me. I'm very excited and nervous. And also for the sponsors to supporting my trip, which was quite expensive and long. Okay, so you know that the Mediterranean Sea is this semi-closed sea between Europe and Africa. I think it deserves some attention, not only because it holds 2.5 million kilometers and around still 3% of the world ocean, but also it fits in the geography of the states. As Brenda told you before, it was completely neglected, as many other marginal seas, in the first estimation on anthropogenic carbon storage in the ocean by Sabine and collaborators. But as I will tell you, it is beautiful, although affected by many problems. You see that the Mediterranean Sea was the cradle of several civilizations, the Greeks, the Romans, the Phoenicians. The Romans used to call it Mare Nostrum, our sea. And now it holds 7% of the world trade. And, 7% of the world population, as you can see here. And most of the people live on the coastal area and in cities. It holds very beautiful cultural heritage sites. That's why it is visited by 30% of the global tourists. This is really, really high. So can you imagine the human pressure here? In addition, I would like to stress that there is a huge difference between the northern countries and in Europe and the North African and Middle East countries. There are socioeconomic, demographic, political differences that make a difference in between those shores, and they also are the reasons for many conflicts. Please remember that in this area there is a migratory crisis. It's full of refugees and immigrants crossing from one shore to the other, and some of them die in the way. In addition, the Mediterranean is a climate hotspot. Everything is exacerbated here, all the changes. It warms faster in the earth and in the water. The sea level is going faster than in the rest of the world. The precipitations are reducing, and fires are also quite extended. Everything is more intense and frequent in this marginal sea area. In addition, not only we have climate stressors, but also the human pressure. As I told you, the Mediterranean Sea air is affected by pollution, with a higher content of particles, ozone, and again CO2 in the area, but also the soil, the water, are especially affected by fertilizers, and also marine litter is full of plastics. So to sum up, the Mediterranean Sea ecosystems and humans can be considered at siege, and the Med Sea can be used as a model for patterns that will be soon experienced in the global ocean. So hopefully any mitigation or adaptation or conservation measures here implemented can be exported as well, elsewhere. So I'm going to start with the general physical oceanography, because it is absolutely essential to understand the biochemistry in this region, which is very unique because it's very different from other areas. And in fact, the whole basin is considered as a laboratory because changes occur, or processes occurring in the global ocean occur here in a much smaller temporal and spatial scale. That's why we can observe them in a human lifetime. But in fact, the Mediterranean Sea can be divided in different subbasins: the Western, the Tyrrhenian, the Ionian, the Adriatic, the Levantine Sea, that function and have different characteristics. So in fact, there is no one lab, but several basin labs. I'm going to start with some general description about the physics. The Med Sea behaves has an antiestuarine circulation driven by the air sea flux. Is an evaporative basin where Atlantic seawater gets in through the Strait of Gibraltar and is modified, gaining salinity and density, and then sinks in the Levantine area, forming Levantine intermediate water that is exported in the Strait of Gibraltar at depth. Okay, so this is why you can see here a huge, quite high difference in the surface temperature and salinity, not only east-west, but also north-south. And the same happens in the intermediate layer for Levantine intermediate water. You see the great increase in temperature and salinity. We have this open cell of antiestuarine circulation of Atlantic water going in and coming out as Levantine intermediate water modified with a very high salinity. But we also have formation of deep waters in each of the different subbasins. You can see here in this map in red the formation of intermediate waters. The main one is Levantine intermediate water that is forming the Levantine Sea, but we also have formation of intermediate waters in the Western Mediterranean by self-dense cascading. Other important areas for the formation of deep waters are by open ocean convection are the Western Mediterranean, the Gulf of Lyon, and in the Western and in the Eastern we have the formation of deep waters in the Adriatic and also in the Cretan Sea. This is why I tell you that the Med Sea is unique as a whole, but with different subbasins functioning differently and with different characteristics for the water masses. In addition, what happens in the Mediterranean Sea does not only stay in the Med Sea, because as I told you, the Mediterranean overflow water comes out through the Strait of Gibraltar and it spreads all over the northeast Atlantic, as you can see here in the salinity signal. This is the salinity, but it also has a signature for the biochemical properties. So any changes occurring in the Mediterranean Sea will finally get into the North Atlantic, affecting the circulation and the biogeochemistry signatures. So long-term changes are occurring. You can see here how the Mediterranean surface is warming and also getting more salty, especially in the eastern part, because here you also need to take into account not only the evaporation, but the damming of the Nile and the reduction of the of inputs from the Black Sea that also have an impact on the biogeochemistry. Here you can see as well that warming is also happening in intermediate waters. You see the stripes for different areas, like in the Eastern Ionian, Sicily Strait, Gulf of Lyon, and in the deep waters of the Western Mediterranean. I would like to know that, take a look at this 30-year time series in the Sicily Strait, led by Katrin Schroeder. This is a choke point because it connects from the eastern and the western part. And you can see that increase in temperature and salinity driven by general warming, but also by sudden changes in the formation of intermediate waters in the eastern Mediterranean. Unfortunately, there are no biogeochemical time series as long as this one. So the Mediterranean has suffered abrupt changes. There is a change in the formation rates of intermediate and deep waters, especially after the 90s, as you can see here. And I'm going to explain you with these diagrams what has happened. Before the 90s, the Mediterranean Sea was thought to be in a steady state, where you had the main water formation for the eastern part in the Adriatic and in the western part in the Gulf of Lyon. But in the 95, around those years, it appeared the Eastern Mediterranean transient, where the main water formation area changed and was occurred in the Cretan Sea. Those waters completely overflow from the Cretan Sea into the deep Eastern Mediterranean, completely changing the distribution, for example, for salinity, where waters get much more warmer and with a higher salinity. Another big change, but in the Western Mediterranean, occurred in 2005, where a very big event of open convection driven by the very huge Earthsea fluxes. And here, there was a massive formation of deep waters in the Gulf of Lyon that completely changed the deep water characteristics for temperature and salinity. And you can see here how the deep water got warmer and saltier, and this water elevated completely the water column in this area. After the 2010s, then again the Adriatic Sea took over the formation of deep waters in the Eastern Mediterranean, and right now this is the current state. So you see that all these changes will affect the distribution of biochemical properties, but unfortunately we don't have as much data to show them as with the physical. With regard to the biochemistry, one of the main things to say is that the Mediterranean Sea is a desert. The clear waters come because there is no much productivity, not because they are so clean, because there is quite pollution in terms of metals and so on. So you can see here the gradient for chlorophyll, and because there is no much primary production, there is no much organic matter to be mineralized, and the oxygen in the whole water column is really high, both in the eastern and in the western. In the western, you can see this minimum here or maximum of AOM, because here the mineralization is higher because the productivity is a little bit higher compared to the eastern Mediterranean. Another strong gradient you can find is for inorganic nutrients. One of the concentrations are really low, especially for phosphate in the eastern Mediterranean. And the reason for this, well, in fact, measuring nutrients in this basin is really difficult because the concentrations, as you can see here, are really low all over the basins. And the explanation for this starvation in nutrients is because of the anti-estuarine circulation that is introducing organic nutrients with Atlantic water that get, and also some inorganic nutrients that get consumed by primary producers. And when this water gets down and is getting out with 11 intermediate water, in fact, all the nutrients that are being remineralized, they are flushed out of the Mediterranean, okay? So this is the main reason to explain the oligotrophy in these basins. Another fact for the Med Sea is the very high nitrogen to phosphorus ratio. And after many debates, the main reason to explain this fact is because there is an imbalance between the high atmospheric and terrestrial input of matter with a very high N2P ratio that is not compensated by the nitrification, because remember that this basin is very well oxygenated. So it's this imbalance between processes that makes the Med Sea an anomalous area with N2P ratio very high. With regard to organic matter, this is the distribution of DOC from west to east, and values compared with the Atlantic, but you can see an enrichment of DOC towards the east, and this is because mainly bacterial activity is limited by phosphorus, so DOC concentrates here. In fact, the Mediterranean Sea can be considered a reactor for organic matter, where it gets transformed from having a more marine origin in the Atlantic, in the Gibraltar Strait, to having a more terrestrial origin within the Mediterranean and getting out with the Mediterranean outflow. Here you can see a degradation index for organic matter, and you can see it indicates how organic matter is reworked and transformed, and you see higher indexes for the deep waters, and how the index also increases with the Levantine intermediate water that comes out with the Mediterranean outflow. With regard to the CO2 variables that are my favorite subject, you can see here a plot for Alkalinity and DIC in gray Pacific waters, in white Atlantic waters, and in colors the different subbasins for the Mediterranean. And you see that the huge, very high alkalinity values for waters in the Aegean Sea and also in the Adriatic. In the Aegean Sea is because of the input from the Black Sea, but in the Adriatic is from the input from the rivers. And as well, you see that the pH values that you can see are high, between 8 and 8.2 compared to the rest of the Atlantic and Pacific basins. but the PCO2 is lower. Looking again at the vertical distributions, you can see how again the west-east gradient for the different properties, DIC, alkalinity, this is pH, and this is omega aragonite and the Riedel factor. There is a huge difference between the basins, west-east, but also north-south, that I'm not showing here. And it tells you that the Mediterranean Sea, especially the eastern part, with a low Riedel factor and high alkalinity, is able to absorb, keep pace with increase of CO2 in the atmosphere much better than, for example, the Western or the Atlantic Ocean. But this ability to capture anthropogenic carbon doesn't mean that it will be much more acidified, because the alkalinity will compensate for this absorption of anthropogenic carbon. And here you can see a nice plot for CFC12 distribution, and you see the higher concentration is deep waters. Oh, okay. Which is related to this formation of huge amounts of deep waters in the eastern and in the western that introduce and ventilate the Mediterranean Sea from the bottom. So as Brenda was saying before, estimating anthropogenic carbon in the Mediterranean Sea is a matter of controversy, because some methods cannot be applied. For example, the back calculation method by Gruber cannot be applied because we don't have the preform conditions, and mixing is really important. The TROCA method is also another back calculation technique, but it seems to be overestimating the anthropogenic carbon, because the TTD method based on CFCs gives much smaller values for anthropogenic carbon. In fact, the TTD method is also could be also an overestimate for anthropogenic carbon, because mixing here in the Med Sea is really an issue. So from a model study, you can see the difference between the change in pH for the TTD and for the model. In the upper water column, more or less, they compare, but not in the deep part. That's why we need data to get values for these changes. But it's quite difficult to get the data in a friendly manner. That's, for example, Brendan was also saying so. GloDAP is a beautiful data product, and I'm trying to produce and release this data product, CARIMET, carbon tracers and ancillary data in the Mediterranean Sea, gathering all this data you can see here in the map and all over the years. From 1976, Frank Liero performed the first cruise in the Western Mediterranean, and I managed to get the data to 2018 for our last cruise, crossing the whole Mediterranean. So hopefully it will be released in 2024 to be used by the community to really assess which are the changes in the Med Sea. So we really need more data about CO2. There are some multidisciplinary time series, especially, of course, led by European countries, and you can find more information in the Goa or Med Sea Hub. And also, we need the basin-wide view for what is happening in the water masses of the Mediterranean Sea, and we are trying to sustain the Med Sea program, where we cross every 7 to 8 years the whole Mediterranean from east to west, and every 5 to 6 years north-south. But I can tell you it's really difficult to get permission, the ship, the funding. And with regard to the future of the Mediterranean Sea, changes are already occurring, not only because of climate, but because of human pressure. Different models, all the models predict, of course, a temperature increase, probably a reduction or increase, not clear about in the mixed layer depth. and a reduction in the returning circulation, and of course a reduction an increase in the evaporation, so increasing salinity. So at the end, that will have an impact in the biogeochemistry, but it's not really clear. So I encourage you to visit Marco's Reale poster, which is about where is Marco? Which is about future projections for the biochemistry in the Mediterranean Sea. So I would like to just conclude or say that the Mediterranean Sea is a hot spot for many things. We need science, but we also need, for getting proper science, diplomacy, cooperation, inclusion, and capacity building, because remember that there is a huge difference between both shores that sometimes they limit and constrain our capacity to observe the Mediterranean Sea. So this is all. If you have enjoyed buying a book, this is a review of the oceanography in the Mediterranean Sea. And just I would like to say thank you to some acknowledgement to Harry Biden, who was a real inspiration for me and for many others working in the Med Sea. Tosse Tanua for giving me the first opportunity to work in a Med Sea cruise in 2011. And Katrin, Jacopo for the confidence on writing the chapter on the book. Many colleagues collaborated, and of course, my group, Innocent in Organic Carbon Oceanography, that that nobody can do anything alone in oceanography. And that's it. Thank you.
Great. Thank you, Martha. So the next speaker is Kitak Lee, and he'll be talking about East Asian marginal seas, and Kitak's from Korea.
Good morning. I'd like to thank the session organizers for inviting me to this great workshop. My talk is primarily based upon the work that my group has been involved in for the last 10 years. I'm going to start my presentation with the geography of the Western Pacific marginal seas, starting from the higher latitude, from Hawaii Sea, moving southward, Yellow Sea, and East China Sea, and the East Sea surrounded by Korea, and Russia, and Japan. For the next 5 minutes, I'm going to briefly talk about the major drivers for changes in the carbon and nitrate cycles in the marginal seas of the Western Pacific. First driver is the extreme seasonal temperature differences. The East Asia monsoons result in a summer and winter temperature difference ranging from 15 degrees Celsius in the East China Sea, and then as much as about even greater than 25 degrees Celsius in the upper part of the Yellow Sea. So it's 20 degrees means it can changes a surface water PCO2 as much as about 300 ppm. So this could be one of the major drivers for surface PCO2 dynamics. Second driver is that this region is very highly productive. In terms of chlorophyll concentration, the phytoplankton biomass in these regions is approximately 5 to even 10 times greater than the open ocean biomass. So this high biomass must be related to the very high input of the nitrate from the continent. This could have a profound impact on the carbon dynamics in the marginal seas of the Western Pacific. Third factor, this is the emerging factor, is the increasing input of the anthropogenic nitrate into the regions. The panel on the left, upper one, is the Chinese ammonia emissions for the last 60 years. It has been tripled over the last 60 years, and the ammonia emissions appears to be stabilized for more recent years. And lower panel shows Chinese NOx emissions associated with a fossil fuel use. And the NOx emission has been increased by tenfold. And then it reaches the maximum at around 2010, and then it began to decrease for the past 10 years. Once the anthropogenic nitrogen compound released into the atmosphere, western wind in this region of the Pacific carry them eastward. During this long-range transport, the nitrogen compound, nitrate, and ammonium ions enter the ocean through a dry and wet depositions. So we tried to identify the anthropogenic nitrogen signals in marine environment. We used the historical data collected from early 1980s up to very recent years, using probably more than 200,000 data points. Covers almost everywhere except northern part of East Sea. Probably none of us can access to the Russian territory. And also, if you look at the North Korea, even Russians can collect any data from North Korea. And at each point, we calculated N star. N star is a measure of nitrate excess or nitrate deficit. This is an extremely simple concept. It was introduced by Nick Gruber and Ray Sarmiento many years ago, back in 1997, I believe, in GBC. And this is the difference in measured nitrate concentration and the calculated nitrate concentration based upon the phosphorus concentration multiplied by Redfield stoichiometry. There are two internal biological processes. Here is the photosynthesis and then respiration. These two processes occur in a Redfield stoichiometry within the internal the interior of the oceans. They do not change the seawater N star values. There are two additional processes, nitrogen fixation and the input of the anthropogenic nitrate into the ocean. These two processes contribute to an increase in N star values in seawaters. Our measurement on nitrogen fixation activities in waters around Korea turned out to be these factors play a very minor role in contributing the N star increase. So any increase in seawater N star changes must be related to the input of the anthropogenic nitrate. So here is the figure in the center. While yellow and red colors indicate the increase in the nitrate concentration relative to phosphorus. So our data indicates that in almost everywhere in the northwestern marginal seas, nitrate concentration increase. In the increase of the nitrate concentration relative to phosphorus, the rate of increase, we found that the highest rate of increase in waters close to the Chinese continent, particularly near the mouth of Changjiang River, and the rate progressively decreased moving eastward. And then so we did find small but statistically significant nitrate increase in the Pacific coast of Japan. Here's a good example. In waters around Jeju Island, south of Korean Peninsula, there has been a considerable increase in nitrate concentration in blue, starting from a couple of micromolars in the mid-1980s, and then it reaches up to about 5-6 micromolars, with a lot of internal variability. However, phosphorus concentration approximately remained constant during the same period of time. So N-star trend pretty much follows the trend of the nitrate concentration. We did find similar increasing trend in N-star in OIC and also Chinese part of the Yellow Sea. The input of anthropogenic nitrate into these marginal seas has progressively transformed the region from being a nitrate limited in blue to a phosphorus limited in red. There are two take-home messages out of this analysis. First one is degree of nitrate limitation has reduced over time. If you look at the color, dark blue means a high degree of nitrate limitation compared to a light blue showing a limitation actually decreased. And second take-home message is the area of P limitations in red color has increased over time, particularly in area located in the downstream of Changjiang River. As I forgot her name, and then Changjiang River discharge NP ratio is over 100. So you actually put out tremendous amount of excess nitrate into the East China Sea, and they did carry all the way up to the Korean coast, south of the Korean Peninsula. Okay. Fourth factor is the crucial current intrusion into the East China Sea. Crucial water very rich in phosphorus relative to nitrate. So we did find some anti-correlation between East Sea N-star values and crucial water intrusions. Greater the crucial water intrusion moving southward, a lower the seawater N-star values in the East China Sea. So, but we did not find any directionality of this the influence. So my guess is that impact of this could be limited. I mean, it can influence intraannual variability, but I don't think it can change the. It has an impact of on the long-term trend of the N-star values in the East China Sea. So, both carbon and nitrogen cycles are very tightly coupled through the biological activity associated with a increasing input of the nitrate into the oceans. So we try to evaluate how nitrate input changes the carbon dynamics in this region of the Pacific Oceans. We primarily used the data that we have been collected from waters around Korean Peninsula since 2015. This is a still ongoing project, 4 times per year from February up to about October. So it pretty much covers the entire season. And we measured both alkalinity and the DIC, and we just calculated other carbon parameters. To complement this data, my colleagues from Handong University, Weidong Zhai, provided the data collected from the Chinese part of the Yellow Sea and the East China Sea, and also Bohai Sea. So we first look at the annual net carbon uptake by this entire basins. Direct measurement point to a very strong sink of the atmosphere CO2 by the East China Sea, and to a moderate sink by the Yellow Sea and Bohai Sea. So strong sink directly under the influence of the Changjiang River plumb. It's moving in a northeast direction, particularly during the summertime. And then annual average mean carbon uptake is about 1.3 moles carbon per meter square per year. It's going to be roughly about 2 times greater than the global average of coastal ocean, which is tabulated by the Minandai last year. But the status is quite diverse across the different part of the coastal regions. I tried to identify the major factors which makes the region becomes very strong to moderate sink of the atmospheric CO2. We plotted seasonal surface PCO2 distribution from winter to autumn, and then pre-bloom to the post-blooms. Surface PCO2 values for winter and spring are considerably lower than the atmospheric CO2, by about 50 to 60 microatmospheres. As season progresses from spring to summer and autumn, the PCO2 values increase to levels that are very close to the atmospheric CO2. There are two processes that they can changes the surface water PCO2 changes over season. One is probably very high production of organic matter and also temperature increase. I'd like to evaluate the relative contribution of these two factors to surface PCO2 dynamics. Here are a very typical surface PCO2 changes shown in black. This is the direct, actually calculated from the alkalinity and DIC, and compared with a temperature-driven PCO2 in red. If you look at these two profiles, 60 to 80% of temperature-driven PCO2 changes were offset by biological activity during the warming period. During the cooling period from summer to winter, surface PCO2 generally follows temperature-driven PCO2, but only, the surface PCO2 only, not as much as the PCO2 changes expected by the temperature changes. The reason for that is during the cooling period, some carbon-rich subsurface water probably being brought to the surface because this water depth is only about 50 to 80 meters, somewhere in that range. There's an exception to the East China Sea. During the cooling period, surface PCO2 very much follows the expected PCO2 changes due to temperature. So the final thing that I like to try is the reconstruction of seasonal amplitude of carbon parameters, particularly pH and saturation state with respect to aragonite. The reason why I do that is the organisms living in the coastal regions and marginal seas are very sensitive to the both mean state of the ocean acidification and also the magnitude of pH changes over seasons. So, if we try to reconstruct the seasonal amplitude of the pH and omega values using the historical data of surface the nitrate concentration increase in the Yellow Sea, and also we use a historical data of near bottom water oxygen consumption rate let me give you the justification that I tried this method. I used the calculation is that depends on the two assumptions. First one is surface nitrate concentration increase can reflect a post-bloom surface DIC concentration resulting from the complete utilization of the nitrate. So you can actually calculate a surface post-bloom surface DIC concentration changes over about 20, 30-year period of time. To estimate the pre-bloom surface DIC concentration, we use a bottom order oxygen concentration changes. The rationale is as follows. In a summertime, a very stabilized yellow sea, bottom order continue to accumulate the DIC because the increasing oxygen accumulation means you can translate this value into a DIC concentration changes in the bottom waters. As mixing intensifies during the cooling period, these carbon-rich waters eventually rises to the surface, most likely reflects a pre-bloom DIC concentrations. Based upon these, the predicted the DIC concentration conjunction with a contemporary alkalinity, we can calculate the pH and saturation state of seawater with omega. And then we did find the amplitude of the pH over season progressively decrease as opposed to an increasing trend of omega over seasons. And then since 2015, the amplitude remained constant for both pH and omega, telling us that probably it reflects a rather stabilized the input of the nitrate compound into the ocean waters. So that means you really don't biology, just activity has remained constant since 2015. This is just not perfect, but it's a reasonable. The result probably indicates that this constant means input of the nitrate probably remained constant, and the activity of the biology most likely remained constant. Okay, let me recap my talk. Input of the anthropocene nitrate into the regions is progressively transformed the region from being nitrate limited to phosphorus limited, increasing productivity, and also increased a remineralization of organic matter near bottom, and then produced a very contrasting seasonal amplitude of pH and omega in this part of the marginal sea. That's it. Thank you very much.
Great. So our next speaker is Zhangxian Ouyang, and now we are moving to the polar ocean. Arctic Sea, make a wave.
Hello, everyone. My name is Zhangxian Ouyang. I'm a postdoc working with Dr. Weijing Cai at the University of Delaware. I want to say thank you to our session organizer, Xinping, Emily, Matt, for giving me this opportunity to talk about our recent research work in the Arctic Ocean. It's been a great honor to be here for real. I will start my talk with a brief summary of the environmental changes happening in the Arctic Ocean, just to give you a better idea about what is the current state of our changing Arctic. And then I will focus on talking about the carbon cycle changes in the Arctic Ocean from the surface water to to the water column. Let's start with warming and the sea ice loss. As we know, the Arctic Ocean is one of the regions, is the most sensitive region on Earth in response to the climate change. Since 1980s, the Arctic temperature increases twice fast than the global average, which is known as Arctic amplification. As a consequence, the summer sea ice extent has decreased by more than 35% in the same period. And these two trends are likely to continue in the next few decades as this ice albedo feedback keeps working. Warming and sea ice retreat as two major drivers also have substantial impact on the land ecosystem. One direct consequence is coastal erosion and permafrost thawing. The study has suggested that the annual erosion rate along the permafrost coast in the Alaska, Canada, and the Siberian has doubled in recent decades. The rate can be as high as 20 to 40 meters per decade. And this rate is expected to accelerate in the future because the sea level rise and extreme heating events in the Arctic. For the permafrost thawing, it has many biogeochemical and climatical consequences. For example, the greenhouse gases methane and the CO2 can be released from the land to the atmosphere. The nutrient and the organic carbons stored in the permafrost can be released to the nearshore region, which further changes the prime production and enhances the sediment flux. In terms of the prime production, a recent model study by Ha et al. 2021 has quantified that the nutrients from river and the coast erosion can contribute about 1/3 primary production of the total primary production in the Arctic. And if you look at this map, the nutrients from the land has a big impact on the shelf sea. And based on the remote sensing product, the researchers have also reported the NPP for the entire Arctic Ocean has increased by 57% in the past 2 decades. And they suggested that for the first decade, the sea ice loss with a higher light availability was the main driver for NPP increase. For the second decade, the biomass increase is the main driver. And look at this map, you can see the largest increase biomass was observed in 2 inflow shelves. One is the Barents Sea and another is Chukchi Sea. And to compare these 2 maps, you can see, although all the Arctic marginal sea has an increased prime production, but the nutrient driver is different in different areas. Next, let's talk about the carbon cycle changes in the Arctic Ocean. Our research is mainly focused on the Western Arctic Ocean. I marked as this red area, which includes one inflow shelf, the Chukchi Shelf, and one river-dominant shelf, the Baltic Sea, here, and open basin, the Canada Basin. And I use this diagram to summarize the physical and the biogeochemical setting for this region. Let's first look at the change at the surface. The first question we want to address is As the sea ice retreats from the shelf into the central basin, in the Arctic Ocean can increase CO2 sink. To answer this question, we need to first understand the distribution of the sea surface PCO2 in this area and examine the long-term trend, how the PCO2 change with time. So we compile a data set and plot all data together. A very unique spatial distribution appears. We had a low PCO2 on the shelf and high PCO2 in the basin. If you take a closer look at this area, there is a clear low to high transition front just along the shelf break. What is the mechanism control this distribution pattern? We attribute it to the influence of different water mass. On the Chukchi shelf, it's a Pacific water dominant, which bring a lot of nutrients, heat onto the shelf. The nutrients can support a strong prime production there and keep the PCO2 low. But this water does not go across the shelf break directly. Instead, it either subducted into the subsurface, follow the isopicanal, or turn the direction to the west or the east, depends on the wind condition. And this is why we didn't see low PCO2 in the Canada basin just across the shelf break. And on the other hand, when the sea ice melts in the Canada basin, this fresh water is floating at the very top surface. That will strengthen the stratification and limit the nutrient supply from the subsurface. Without nutrient supply, the prime production at the surface is very weak and cannot last for a long time. So the PCO2 there can increase very quickly due to the CO2 uptake from the atmosphere. Let's take a look at the trend. We separate this area into, we focus on these two most contrasting areas. On the Chukchi shelf, the PCO2 doesn't change too much. The trend is not significant. But consider the atmosphere CO2 continuous increase. The delta PCO2 actually increase as well with the time. So we suggest that the Chukchi shelf is an increased CO2 sink. In contrast, the PCO2 in the Canada basin significantly increased at a very large rate. The rate is about 4.6 microatmosphere per year, which is more than 2 times atmospheric CO2 rise. As a result, the delta PCO2 quickly decreased. That suggests the Canada basin is a decreasing CO2 sink. And if this trend continues, the delta PCO2 will decrease to near zero in the next 10 years. That means the Canada basin will not take extra CO2 from the atmosphere. And another question is why PCO2 in the Canada basin increased so fast? And what is the dominant driver for that? So we decompose the PCO2 increase into 2 components. One is the thermal component, which is driven by the long-term change in SST, and another is non-thermal component, which is driven by other factors like DIC, alkalinity, and salinity change. And we found that the non-thermal component can contribute about 88% of the total PCO2 increase rate. So we propose a mechanism to explain what we observed. It is we call it a synergistic effect of 3 drivers, 3 processes: ice melting, warming, and the CO2 uptake. In the early 1990s, most area in the Canada basin is covered by ice, so the gas exchange is very limited, and the surface water is, the temperature, the warming is also weak. In the recent year, larger area become ice-free. It allows more CO2 being taken up from the atmosphere. And another process is dilution by melt water. The melted water dilutes the surface seawater, give it a lower PCO2 at the initial PCO2 after the ice melts. And without ice cover, a lower PCO2 will allow a larger CO2 uptake, which leads to a large DIC increase. As a result of DIC increase at the surface, pH and omega also decrease. This is not something surprising, but what makes us very surprised is the rate. We quantified that from the 1994 to 2020, the surface ocean acidification rate in the Canada basin actually is about 3 to 4 times higher than any other ocean basins. And also notice a strong linear relationship between the summer sea ice extent and the pH and omega. The lower sea ice, lower omega, lower pH. And we proposed a mechanism called ice melting-driven acidification to explain what we observed. Simply speaking, when the sea ice, massive sea ice melt, this melt water dilute surface seawater, decrease the salinity, alkalinity, and the buffer capacity, make this surface water more sensitive, more vulnerable to DIC perturbation. That means if just a small amount DIC added to this water, surface water, it will lead to a large pH and omega decrease. And this is a very interesting study model, just published in last year by all et al. And they found the annual high in PCO2 could shift from winter to summer. From winter to summer, yes. Look at this map. This seasonality of the PCO2. The blue line is the PCO2 seasonality at present, shifted to the yellow dashed line in the future. And they emphasize this intensified warming effects will become the dominant driver for the PCO2 variation in the future summer months. And look at this map, you will see this driver change from the non-thermal component to thermal component are more has a larger impact on the Arctic Gulf Sea, or we call it marginal sea. This is really bad news for the organism living in this marginal sea. Okay, next, let's talk about the changes in the water column. Before I show the data, I would like to introduce the typical water column structure in the Western Arctic Ocean. Compared to the east side, west side has, the structure is more complicated. We have a surface mixing layer, Pacific summer water, and the Pacific winter water, and Atlantic water at the bottom. And I want to have your attention to look at this Pacific winter water layer. We are very interested in this layer because it always associates with a DIC maximal, pH minimal, and omega minimal. In other words, this water, Pacific winter water, is a forerunner for the subsurface ocean acidification in Arctic. Another model also predicts the lowest pH and omega value in the future will appear at subsurface, and marked with a red arrow. And I think this is also associated with the Pacific winter water. Okay, I cannot see. Next, we talk about how the Pacific winter water intensifies the subsurface acidification. Based on the observation from 1994 to 2010, we noticed that the undersaturated water with regard to the omega expands to deeper and further northward from 1994 to 2010. You can tell the change by, well, the blue area changes. And we also quantified that this blue area, the water, undersaturated water has increased by 3 times. And that is because the increase of the Pacific winter water intrusion to the deeper and further northward. And we noticed there is a linear relationship between the fraction of the Pacific winter water and the omega. Larger fraction of Pacific water will give a lower omega. And we conclude that the increased Pacific winter water is a primary driver for the saturated water expansion and subsurface acidification. The story doesn't end here. Someone may ask why the Pacific winter water has the highest DIC and what is the carbon source for that. Actually, there are several papers already published, and they use multiple tracers to answer this question. They all suggest that the carbon from the shelf, transport from the shelf to the basin, is a main contributor of the DIC to Pacific winter water. But the remaining question is this carbon comes from the land or it's a marine origin? If it comes from land, it's possibly delivered from the river or released from the permafrost soil or coastal erosion, or it's possibly a mixture of both signals. And to carbon source partitioning is very challenging in this area because if you look at this end member plot of the delta carbon 13 POC, they all overlapping with each other. So really hard to differentiate the terrestrial organic carbon from the marine organic carbon. So what we do, we try to use a different approach. We use salinity delta carbon 13 nutrients and to build a 4N member mixing model for DIC and the DIC delta carbon 13. The goal is we trying to quantify how much DIC change and how much delta carbon 13 change is due to the primary biological process, basically the respiration. What we found is the organic carbon in the Pacific winter water on the shelf is more like marine origin because the signal is more close to the marine phytoplankton. But this signal change to be more deplete, more negative in the basin when this water partial transition from the shelf to the basin. This is a very surprise to me because check the sediment record of the POC delta carbon 13. We cannot find such a negative value from the Chakti shelf. It's more like from this way. from above the sea. And I don't know how to explain this until I come across these two papers. From the physical oceanographic perspective, they provide one explanation. When this center of the gyro shift from the south north to the east south, they found the winter water formed at the eastern above the sea shelf can make a contribution to the winter water in the Canada basin, and the organic carbon delivered from the land can go this way into the Canada basin. Okay, that's all what I want to cover. Thank you.
All right, so the last but not least talk for the Gulf of Mexico. Next speaker is Andrea Quiloha, and she is actually dialing in from Hawaii. That's quite a few hours after me, earlier in the morning. And Gulf of Mexico there, please make way.
Aloha, everyone, and mahalo to the organizers of this session. I will be discussing surface water CO2 variability in the Gulf of Mexico. And you might be thinking to yourself, why is this Hawaiian girl talking about the Gulf of Mexico? But I actually did my PhD at Texas A&M, so I was really fortunate to learn about a new ocean beyond the Pacific. So I'll be discussing the kind of general spatial, seasonal, and long-term trends in CO2. Okay, so the Gulf of Mexico is a critical component of both regional and national economic stability. There are a number of important fisheries in the Gulf of Mexico, like shellfish fisheries, and seafood landings in the Gulf are almost 20% of total US landings. So the health of this ecosystem is really important in providing and supporting jobs in the region, but also for providing food to people across the US. The Gulf of Mexico serves as an important drainage for a number of different rivers, including the Mississippi and Atchafalaya rivers, which drain about 40% of the contiguous United States. And then the Gulf of Mexico hosts a number of important habitats like coral reefs and 50% of the nation's wetlands. Despite its importance, the Gulf of Mexico faces a number of anthropogenic threats, and I'm just going to talk about a few of those today. So there's a number of different rivers that deliver high nutrient concentrations into the coastal waters. This leads to eutrophication, hypoxia, and coastal acidification. And then we also have the global climate change stressors that affect the entire region: ocean acidification, warming, and episodic storm events. Hypoxia was first reported in the Gulf in the 1970s, but the sedimentary records suggest that low oxygen became a problem in the early 1900s and then accelerated around the 1960s due to urbanization and industrialization. The northern Gulf of Mexico, hypoxic or dead zone, is the second largest hypoxic zone in the world, and it's monitored seasonally, so we know quite a bit about this dead zone. This figure is from the summer 2022 hypoxic zone, and we define hypoxia as dissolved oxygen concentrations less than 2 milligrams per liter, which is shown by this black contour line here. And the Mississippi and Atchafalaya rivers are also shown on this map. These rivers deliver high nutrient concentrations, particularly nitrate, to the coastal waters. This stimulates productivity, so peak discharge is in the spring, and then in the summertime, productivity continues, but we get warming and vertical stratification of the water column. And as that organic matter sinks to the bottom, it respires. And because we have stratification, that prevents reoxygenation at depth. So it's the combination of these processes that leads to hypoxia. In addition to hypoxia, the flux of nutrients from these rivers also induces coastal acidification. So this is a figure from Laurent et al. 2017. It's generated from a biogeochemical model showing pH on the top bottom water pH in the top figure and the saturation state of aragonite on the bottom figure. And within the hypoxic zone, pH can get below 7.7 and saturation states can be below 2, which is still supersaturated, but calcifying organisms can dissolve in saturation states greater than 1. So it's these numbers are still pretty alarming. More recently, it's been shown that episodic storm events can also lead to coastal acidification. In August 2017, Hurricane Harvey made landfall in Texas as a Category 4 hurricane and actually stalled over Southeast Texas for about 4 days, releasing massive amounts of rainfall up to 100 centimeters in some areas and causing catastrophic flooding. And a lot of these floodwaters were released into Galveston Bay, which I'm showing here in this red square. So Hicks et al. Conducted a study in which they looked at the effects of these floodwaters on the carbonate chemistry of Galveston Bay. The top figure is showing pH, and the bottom figure is showing the saturation state of aragonite, and this is over approximately 1 year. The first symbol is pre-Harvey conditions. The orange line is during Harvey, so that's a 4-day period. And then the first symbol after that line is about 2 weeks after Harvey. So they did see a significant acidification event after Harvey, where pH dropped below 7.6, and all stations within Galveston Bay were undersaturated with respect to aragonite and calcite. Galveston Bay is actually a really important place for oyster reefs. So you can imagine that was detrimental to the reefs there. And then it took about 2 months for the carbonate chemistry to return to pre-Harvey conditions. With all of these stressors that can impact the carbonate system and cause acidification, sustained observations are really important for providing information about long-term trends in biogeochemistry and how our ecosystems respond to these changes. We have a number of open ocean time series stations located across the globe that monitor for ocean acidification. And I'm showing you those stations with the red symbols. And these are the PCO2 trends at each of these stations. So they all show the same general trend that as CO2 increases, as atmospheric CO2 increases, so does surface water PCO2. But the Gulf of Mexico really stands out as one of the major regional gaps in our knowledge of ocean acidification. The objective of this study was to synthesize all publicly available underway data to describe the physical and biologically driven surface water PCO2 trends in the Gulf of Mexico. So we downloaded approximately 380,000 measurements of sea surface salinity, temperature, and PCO2 from SOCAT across the years 1996 to 2017. So these are measurements collected by the flow-through systems on ships, and I think we used over 200 cruises. So I just want to acknowledge all of the researchers and technicians and funding agencies who have contributed to the collection of this data. And I'm showing you a map of all of the measurements that we used, and they are color-coded according to season. So winter is dark blue, spring is light blue, pink is summer, and green is fall. And right off the bat, you can see that the majority of measurements were collected in the spring and summer, about 75%. That's because hypoxia drives a lot of the research in the Gulf, and hypoxia is a spring and summer phenomenon. But also it's very cold in the Gulf in the winter, and so nobody wants to go out in a ship, I think. Also, the majority of the measurements, 99%, were collected after 2001. We also lack measurements in the southwestern region, so this region continues to be a gap in our knowledge for CO2 trends. Despite the limitations of the data set, we were able to produce the most comprehensive analyses at the time of spatial, seasonal, and long-term CO2 trends. So here I'm showing seasonal CO2 trends for winter and fall, where blue is low and green is high. We see low CO2 due to cooler temperatures. This is partially offset by enhanced respiration, which we can see in the normalized PCO2 data, which I don't show in this presentation, but it's in the publication if you're interested. And then we do see pockets of high CO2, particularly near the rivers due to riverine inputs and outgassing. In the spring, CO2 increases with warming, but this is offset by an increase in productivity, so that the net effect is lower CO2 relative to atmospheric CO2 across the region. You'll also see near the coast this highly productive area that's driven by the Mississippi and Atchafalaya rivers. We also have a western coastal flowing current from the winter to spring, so that current transports the low CO2 waters near the river mouths westwards toward Texas. And then in the summertime, CO2 increases with temperature. We still continue to see a lot of biological productivity occurring near the river mouths. And what's kind of interesting and unique about the Gulf of Mexico is that western flowing current actually switches east and offshore during the summer. And we also have these eddies that break off of the loop current. And when they impinge upon the shelf, the combination of these processes acts to enhance cross-shelf transport. So they can transport this low CO2 water out into the open ocean, hundreds of kilometers, which has implications for open ocean ecosystems. We also separated the open ocean and coastal ocean data to see if we could detect any long-term CO2 trends. So here I'm showing the open ocean data for years 1996 to 2017. The blue line represents the atmospheric CO2 record, and the gray symbols are the deseasonalized monthly means. We see an increase in surface water PCO2 in the open ocean, approximately 1.2 microatmospheres per year. So this is evidence for ocean acidification in the open ocean Gulf. And if we go back to our figure here, this 1.2 is at the lower end of the other open ocean time series stations, but still compares really well. Detecting an ocean acidification signal in the coastal ocean is really challenging. You need a long-term time series to tease out the ocean acidification signal, which is much smaller than the natural variability of the system, but we attempted to do this anyways. So here I'm showing the coastal ocean data where, again, the blue line represents atmospheric CO2 and the gray symbols represent the deseasonalized monthly means across the years 1996 to 2017. You can see that there is a lot of variability but we do detect a significant increasing trend of 4 microatmospheres per year. This is 3 times faster than the open ocean. We also found an increasing temperature trend. So this temperature trend can account for 65% of the PCO2 trend, and the rest we can attribute potentially to uptake of anthropogenic carbon. We also calculated long-term PCO2 trends for various subregions across the Gulf. So we have 3 coastal regions and 6 open ocean regions, but 2 of these open ocean regions, the Southwest and South Central, didn't have enough data. So we end up with 4 open ocean regions. And I'll start with the open ocean long-term CO2 trends. These are in microatmospheres per year. So I'm looking at the red boxes for the Northwest and Northeast open ocean regions, subregions. These trends are comparable to hot and BATS. And then in the Southeast open ocean, I'm pointing at it. I'm not sure if you guys can see my cursor. But we also found an increasing SST trend. So 25% of the increasing PCO2 trend can be attributed to rising SSTs. And then in the central Gulf of Mexico open ocean, we don't see any long-term trends in SST, salinity, or PCO2, potentially because this region is influenced by the Mississippi and Atchafalaya river systems, which introduces variability that may mask long-term trends. Moving on to the long-term CO2 trends across the coastal subregions, in the northwestern Gulf of Mexico, we do see an increase of approximately 3.2 microatmospheres per year, despite no significant trends in SST or sea surface salinity. So the acidification signal in this region is potentially driven by a decline in the photosynthesis to respiration ratio and or uptake of anthropogenic carbon. On the West Florida shelf, we see increasing sea surface temperatures that can account for approximately 40% of the increasing PCO2 trend of 2.35 microatmospheres per year, and the rest we can attribute to uptake of anthropogenic carbon. And then in the north central Gulf of Mexico, we see the same thing as the open ocean central region. No significant long-term trends in SST, salinity, or CO2. And again, this region is really influenced by the Mississippi and Atchafalaya rivers. And so it's just highly variable in a very productive region. It's also possible that increasing photosynthesis driven by nutrient loading is masking the acidification trend, or that the time series is just not long enough for the ocean acidification signal to emerge. So in conclusion, we've identified seasonal and spatial CO2 trends across the region and find that biological productivity linked to riverine water and temperature seasonality are the primary drivers of PCO2 trends across the region. We've identified significant long-term open ocean and coastal ocean trends, and we find that the coastal ocean is acidifying approximately 3 times faster than the open ocean, with the exception of these kind of central regions. And we're not quite sure why these long-term OA trends within the central regions are masked. And finally, 20 years of underweight data does provide insight into space and time trends, but the establishment of a Gulf of Mexico open ocean time series station that includes carbonate chemistry measurements would be really helpful in calculating long-term CO2 trends with better certainty. And with that, mahalo.
So I'll hand it over to Matt, who's going to facilitate our panel discussion.
Okay, just starting with panel discussion. So I'm from Brazil. Am I the only one cold in this area here? Because it's cold. Okay, so the first question is, what is the best marginal sea? Just kidding. That's not the first question. Let me get the first question, actually. Okay, what is the most important role the marginal seas play in global biogeochemical cycling from your perspective? Anyone?
I would say that the carbon export is the most important task that marginal seas play in global role. Yeah.
Nice. Anyone else?
I can just say for the Arctic marginal sea, as you can see from that two map, the marginal sea, especially for the inflow shelf, the Chukchi Sea and the Barents Sea, they are two largest CO2 sink in the Arctic. And the role of the marginal sea in the Arctic, I think the most important thing is it help the carbon export from the shelf into the basin. Because the residence time at this two inflow shelf is very, very short. So the strong prime production there fix the CO2 from the atmosphere and become the organic carbon can quickly export to the deep ocean.
Thank you. Go ahead.
I think very difficult questions to answer. I've been working on this anthropogenic CO2 uptake by the East Sea of Japan. Overturning circulation within this region is a curve within about 30 to 40 year time period, compared very short relative to the North Atlantic meridional circulations. I think marginal sea, in terms of the oceanic CO2 uptake, like a Mediterranean Sea and the East Sea, can serve as a platform to identify what is the tipping point in terms of the oceanic uptake of the CO2. There's a possibility that when ocean gets warm in the marginal sea, that they respond very rapidly to their uptake rate considerably decreased within a short period of time. So they will tell us that there's a strong implications to the role of the meridional, North Atlantic meridional circulation, how that will change in terms of the oceanic CO2 uptake. When the temperature warms up by 2 degrees, what if East Sea CO2 uptake completely shut down? That clearly tell us that maybe a 2 degrees Celsius increase is a tipping point for the slowing down of the oceanic uptake of the CO2. So maybe I think that's it.
I might be missing the question, and that might be confusing coastal seas and marginal seas. But I do know that the biological productivity in the marginal seas, I don't have any statistics on this, but would have to support a tremendous amount of the kind of fisheries that humans rely on. So I would just kind of point to that as probably really significant.
Andrea, Sophie, do you have anything to add? or no? It's anyone else? No? Okay. Let's go to the second question. So what are the it kind of looks like a show, right? Like I'm asking the participants here, but so what are the major knowledge gaps, okay, both observationally and with respect with modeling, with respect to biogeochemical cycling within marginal seas? And the second question is, where does the research funding need to be focused?
Well, I'm astonished by the amount of data and shown. I was like, my God, if only we had those temporal, those time series in the Med Sea. Well, we will learn a lot, because I think that the Mediterranean Sea is suffering warming, salinification. With the salinification, the alkalinity is increasing, so it's like a way of alkalinity natural alkalinity enhancement. So we'd better get better and more data. And as I try to explain you, it's very difficult because you are running into economic national areas that you are not allowed to cross. And there are so many diplomatic conflicts, especially in the Levantine area, where I tell you there were these abrupt changes in the deep water formation. That is difficult to get the data. And national data from, I don't know, maybe Israel or Lebanon, they don't want to see each other. They don't want to share anything. So it's difficult to gain knowledge on that. So that's why we need to break many barriers and be more, how do you say, open for collaboration, for funding, for capacity building. It's not easy, but we need to do that.
Yeah, I would just add some new insights that haven't been tackled yet, is that also in the marginal seas or the coastal oceans, the new anthropogenic influences that occur, like wind power plants and stuff, is also a knowledge gap we don't know yet, like the influence of them on the on the ecosystem itself, on the biogeochemistry, but also on the general habitat for animals and stuff like this.
I actually would like to add something too. So last year I went to this WCRP, and so part of this question is kind of related. And they said that the land development in global climate modeling is not as developed as atmospheric and oceans. So is there anything that we should be focused when it comes to like marginal seas that we're not focusing right now in relation to ocean modeling, coupling or something?
I'll think about that some more as well, but one thing that I do know, I would say it is an area of focus, but trying to get the ice coverage well represented in models is a real challenge.
I could add something to that too.
Go ahead.
Okay. I don't really know about the other marginal seas, but at least for the Gulf of Mexico, understanding the rivers that contribute to carbonate chemistry in the Gulf, I think is something that we need to do better and include in the models.
Yeah, to add on Andrea's comments, I think we need to better resolve the river forcings, because in global ocean modeling and even for Earth system model, we just consider that what is happening on land is directly rooted to the ocean. But we need to better consider the filtering, the recycling along the land, ocean, aquatic continuum.
And I would say seasonality, it's a really hard part for model, especially in the Arctic Ocean. I have a lot of data shown there, but actually they are all in the summer months. We don't have any, like very few observations in the later fall, early winter, and the early summer, early spring. So far we don't have a complete seasonality of the PCO2 cover in the Arctic Ocean.
So is there where the funding comes in, right?
Actually, I would like to tie that response into the last question as well. It's also arguably an observation gap. And there are a handful of places in the world where there are such frequent cruises that we've been able to show that with access to that kind of data that does fully resolve the kind of annual seasonal cycle, even just 4 times a year or something like that, we can develop algorithms that can really actually resolve that seasonality and capture it very well. So there's a whole lot that we could do if we had better seasonal coverage. And that, again, kind of points to some of the strengths of programs like biogeochemical agro that have the potential to kind of keep going out there much more frequently than we can get ships out there.
Goodness. Really, this is something special. Okay, I'd like to go back to a comment I made to Hongjie regarding groundwater inputs. And the emphasis has been on rivers, and that's just the surface expression of land-sea fluid interactions. And I think it's incredibly important in marginal seas and in the coastal ocean generally to constrain these submarine groundwater inputs. And so that needs to be done in conjunction with the rivers to really fully constrain those kinds of interactions. That's one comment. The other thing I'd like to say is one of the projects I was involved with earlier in my career was one called AMOCEDS, and it was focused on the Amazon shelf. And it was a multidisciplinary and interdisciplinary approach to sampling the system. And there were physical oceanographers, geological oceanographers, biogeochemists, and unfortunately there wasn't a major biological component in the seabed because the biological oceanography program at NSF was funding at the time only manipulative experimental approaches, and they were resistant to involving field research. That aside, my point is when we enter into looking at these complex systems, you really can't do it from one perspective alone. You can't have just one discipline coming in and trying to monitor their particular favorite measurements. You need to look at it as an integrated, complex system. So you need people with different expertise simultaneously examining the system. And so I would urge any plans for future focused research on an area like this to involve multiple disciplines, not just biogeochemists, but other people as well, and to really constrain what's happening as much as possible. So that's my really overall.
I just want to add some consideration to what Martha said before. I'm an ocean biogeochemical modeler. Yes, we really suffer the lack of data in the sense that data are not only important, observation, I would say, are not only important for evaluating the performance of the model, but at the same time, they're also important, for example, just to initialize the model. Okay, if you put the wrong initial condition in your model, you can simulate different dynamics with respect to the reality. If you have no data, you cannot produce eventually also boundary condition for your model. And this is important when you consider the Mediterranean Sea, because at the end it is even enclosed basin. So something is the internal dynamics strongly depends, for example, on the Atlantic Ocean. So we don't have enough observation in the Gibraltar Straits. Probably we will have some problem in simulating the future evolution of the basin. if we do not have enough observation, we cannot simulate properly some phenomena. We cannot proper set some parameterization in the model. And also another important aspect I think that should be considered in the semi-marginal sea is not only the river load. I absolutely agree with the importance of the river load, but also the atmospheric deposition, for example. Because in the future, we expect an increase in the vertical stratification, so which means that we will have a lower injection of nutrients just through the vertical mixing. So in this case, atmospheric deposition can play an important role in changing the future of the marine ecosystem. I'm talking about the Mediterranean Sea because it's what I do, climate modeling and biogeochemical modeling.
Ask a question here. I want to follow up on Bob's point. Like, for example, for lease research, it's very good. We see this shift in dominant nutrient. So follow on that, do we see any shift in the plankton population to a more like phosphorus free rate?
In our paper in limnology and oceanography and Korean fishery agencies, they got a long-term record in a few phytoplankton species, the diatom versus the dinoflagellate. I'm not 100% sure for the validity of the data, but we analyzed those direct observations. It tells us that it appears to be shifting from diatom dominating blooms to a more frequent dinoflagellate blooms, particularly in waters around Korean Peninsula, and also I heard that occur in waters near the Changjiang River. That's the only thing that I can say. Thank you.
Professor Lee, I think you're in the hot seat over there because I have some thoughts to discuss with you. Also a question for you. So talking about tipping point, again, if you look at it from historical perspective, I don't think there's any, oh, sorry, I made a mess. Sorry, I made a mess. In Earth's history, I don't think there's any single time like when marginal seas would totally stop, like being sequestering carbon. And again, if you think of like the dinosaur ages of Cretaceous, that's actually when a lot of our petroleum are coming from the Cretaceous, where the Earth is not only 2 degrees warmer, but it's like 12 degrees or 15 degrees warmer than today. That's a thought to share with you and everybody. But my question for you is, I'm really curious about this, the nitrogen to phosphorus ratio from the Changjiang River. Why is it so high? If it's a fertilizer, why don't people apply like a phosphorus, but just the nitrogen?
The phosphorus is it's a limited resources. The nitrogen, we can synthesize the nitrogen compound. So I know that many terrestrial ecosystems still, when you cultivate something, you need the soil to need the nitrate. So I think, and also what I heard is that the efficiency is really bad. If you throw the nitrate fertilizers into the system, probably 50 more, even greater than 50% are just washed away. So, but the farmers keep using more and more nitrate. So I think that's the one reason why a river system contains very excess nitrate relative to phosphorus. So you got a limited resources. You only have a natural source of phosphorus.
Okay, thank you. Is that unique to Changjiang, or like what about Mississippi?
I think the statements that you made apply globally, but I don't know the specifics of the Mississippi nutrient loading.
I don't know the specifics of the Mississippi there. I'm the Gulf of Mexico person, so but I'll just add to phosphorus binds to soils. It's just not released as easily as nitrate, so that could be another reason why we just see higher nitrate concentrations.
Last question, and it's worth 10 points. Are there marginal seas mitigation or adaptation measures that may lead to a better environmental status or improved sustainable management of resorts or ecosystems? 10 points, people. Who is going to win? What is the best marginal sea?
I don't know if this answers the last question, but I think it's a point that is important to make. The role of groundwater was raised as well, and a lot of the times I think, especially in the modeling world, we're thinking of, you know, whatever comes from land as a source to the ocean, and there's an interaction. If you talk about, you know, groundwater modeling, there's water from the ocean that's going to go in there, and that water is going to have a residence time, that stuff is going to happen, and then that's going to wash off back. You have ghost forests that, you know, water, sea level rise is playing a role, and, you know, something's going to happen to that organic matter that's dying, and that can get washed off at some point. So it's not a fixed boundary, and we should talk more to the land people on how to adapt and how to incorporate these processes that are happen on land, quote unquote, right? Because it's in the water. To how do we go about parameterizing that into our models? How do we go about linking these processes and this dynamical source to the ocean? So I think that's a point that needs to be made if you're talking about like mitigating consequences to marginal seas.
Thank you. 8.5, 8.5. I'm done with my questions.
Okay. How about another round of applause for our speakers? That was awesome.